Welcome. A very good morning to all the respected guests, dignified speakers, respected teachers, and all the dignitaries present here. I am Sakshi Thakkare.
And I am Vanshika Lalwani. We both are from final year School of Pharmacy, CHHINDWARA University, Saikheda. And we extend our delightful welcome to all the dignitaries present here.
As we all know, today is the second day of our international conference, APIIR, that is Approach to Pharmaceutical Insight through Interdisciplinary Research. It's an opportunity for us to come together, gain knowledge, and engage in meaningful conversations. We have several lineup of sessions of our respected speakers and thought leaders who will guide us throughout the journey.
As we know, light is a symbol of joy and prosperity. And to invoke the blessings of Goddess Saraswati, let's begin our today's event by lighting the lamp. Now I request all the dignitaries to come near the dais for lamp lighting. So please everyone proceed. And I request everyone to stand on their respective places.
प्रश्नों की फसलें खिले, जिज्ञासा के बीज भी हर एक के दिल में पले। ज्ञान के दिन हर जले, अज्ञान की रैना ढले।
नित सत्य की हो कामना, गंतव्य नहीं एक राह है।
गुरुकुल की ये संकल्पना, ज्ञान की आराधना, ज्ञान की वो साधना, सत्य की एक खोज है। गुरुकुल की ये संकल्पना, गुरुकुल की ये संकल्पना।
Before proceeding our event, now it's time to welcome our respected guest. So, I would like to request Dr. Sriram Joshi sir, Registrar, Raisoni University, Saikheda, to please welcome Dr. Swarup Lahuti sir by presenting token of love. Thank you so much, sir. Now, I would like to request respected Dr. D.C. Sahu sir, Dean, School of Pharmacy, CHRU, Saikheda, to please welcome Dr. Jay Prakash Shetty sir by presenting token of love. Please, sir. Thank you so much, sir. Now, I request respected Dr. Dhanashri Tumbe ma'am to please welcome Dr. Nitesh Jain sir by presenting token of love. Thank you so much, ma'am. Now I would like to request Amol Theri sir to please welcome Ritesh Khatkar sir by presenting token of love. So, before proceeding our event, let's have a quick review of the motive of our international conference APIIR.
The conference aims to focus on exploring innovative and collaborative approaches in the field of pharmaceuticals. By bringing experts together from various disciplines, the conference aims to foster interdisciplinary research and provides insights into the development, production, and utilization of the pharmaceuticals.
The conference will emphasize the importance of collaborations between different disciplines such as pharmaceutical sciences, biochemistry, pharmacology, biotechnology, and other related subjects. It will highlight how combined expertise from multiple fields can lead to breakthrough in drug discovery, drug knowledge, formulation, and drug delivery. So now I would like to hand over the stage to my colleague Vaibhav and Nikita. So please.
Thank you, Archana and Sakshi. Now, I would like to introduce the first guest speaker of today, Dr. Swarup Lahuti sir.
Sir is professor and head of pharmaceutics department in Y.V. Chauhan College of Pharmacy, Rozabad, Aurangabad, Maharashtra.
He has completed his bachelor's in pharmacy and master's in pharmaceutics from Nagpur University. Sir has teaching experience of 23 years, individual experience of 2 years. In his guidance, more than 80 students have completed their masters and 5 students have completed their PhD.
Sir has 5 patents, 55 research publications, and received 2 international travel grants. Sir is currently elected as Vice President of APIIR Maharashtra. We are highly honored to welcome you, sir, with a big round of applause.
Please. Good morning, everybody. So, at outset, I'm thankful to the principal, registrar, and the management of Raisoni Group of Institutions for giving me opportunity to interact with you all. And the theme of this conference is interdisciplinary, means a blend. And I can see this is a real blend, what I can see a blend of Maharashtra and MP. So this is a blend of Maharashtra and MP that I can observe here. So this is a real interdisciplinary crowd is also there. So some are Marathi speaking, some are Hindi speaking. So you can see the advantage of this interdisciplinary. You can exchange your ideas, you can exchange your thought. And similarly that is occurred into this science also. And recently this interdisciplinary approach is emphasized even by the government. After implementation of NEP 2020, I think maximum crowd is of B.Pharm. How many of you are M.Pharm students? Very good. So it's a blend of B.Pharm and M.Pharm, the faculty members are there. And as you are aware, now NEP 2020 is being implemented. And implemented drastically by the government at all the levels. And government is giving the emphasis that in NEP 2020, if you go to the NEP 2020, there should be an interdisciplinary approach in every aspect. In education also, in research also, everywhere there should be interdisciplinary approach. So I congratulate the Raisoni group for organizing such an international conference on which is a really needed topic in present scenario of NEP 2020. So my topic, start the topic later. So first thing, there is a question you can see in this slide. You can observe the question is there. What is more easy and economical? That is to develop any particular new thing or to modify the existing things. What do you think? Obviously, the modification of existing is always the easy job, economical job, and also the result producing job, because to start from the scratch is a difficult. So rather than that, it has already been existing. So improvement is more important. Improvement is more important. So you can see there's certain small sayings are there. Small changes eventually can bring a big change into any particular thing. So that will also occur with the API molecules. So in the light of this particular question, my topic is, next slide please. This is okay. What? It's working? No problem. Because see, I'm used to a teaching of always moving in the class. Yeah. So that's why I feel that the movement is always essential. What happened? So the to improve is to change and to because any change make it a goods more perfect and perfect. Because nothing is constant in this world. The only thing is constant is a change. So that's why if we change the small things into the API molecule, what drastic difference may occur? So this is a theme of this particular research work. So topic is a drug co-crystals, a novel game-changing approach to alter the properties of API. Again, the game changing, you know that game changing means it will can bring a very big change into the pharmaceutical industry if this is being implemented. So this drug co-crystals, everybody is aware about the crystals, crystallization. We discussed that. Next slide, please. Next slide. So this drug co-crystals is a recent concept which is being introduced into the research therapy. If you take the total highlight of the therapy which is available, आप सभी को मालूम है, that is one is monotherapy, that is single drug is given at a time. There may be a more than one drugs, they are given in different types of dosage form. Third thing is available, the fixed dose combination, that's what the FDCs are there, fixed dose combinations are there. These fixed dose combinations, a particular combination like paracetamol and the diclofenac, fixed dose combinations are there. But what happened? Lots of restrictions that has been occur on these fixed dose combinations because of certain legal issues. Many FDCs they have been prohibited by the Food and Drug Administration. So FDC is another approach, and next approach which may be introduced, which may come as a game-changing scenario, that is drug co-crystals. It is basically a combination of two drugs. Combinations of two drugs, and that will give the advantage of both the drugs simultaneously. Next slide, please. Problem with it? Okay. So if you see the active pharmaceutical substances are there. In case of active pharmaceutical substances, what should be the vital properties of any particular API molecule, or what is the expectation from one particular API molecule? क्या क्या चाहिए API से आपको? What do you expect from API? First thing, what is required? It should have a good potency. It should have good potency is required. Effectiveness is required. Potency is required. Next thing is required good ADME, absorption, distribution, metabolism, excretion properties are there. Then require a good selectivity, because not only potency but selectivity is also important criteria for the action, because it should not show the adverse effect on the other organs of the body. Other than that, also require material properties, because as a formulation scientist, it should have a good compressibility, good flow properties, and good solubility. That is material properties are there. So if combinations of all this you can see, this is a drug molecule where the optimum properties of all that is been selected. Optimum potency, good ADME properties, good material properties, and good selectivity that is selected. And if you see the APIs of the drug substances which are available into the market, they are of different categories. If you see the solid classifications of the API, that may be available as an amorphous form or the crystalline form, as you are aware. Many M.Pharm students might make a work on the conversion of drug into amorphous form. Many M.Pharm students they are doing the work on solubility enhancement techniques. Right or wrong? Correct, look. Solubility enhancement technique. So conversion of crystalline form into amorphous form, maybe a solid dispersion or other techniques you are using. So the amorphous form is there. Amorphous form is more soluble. If you consider crystalline form, it may be a single component system or the multi-component systems. Single component means only one component is there which is present. In multi-component system, it may be one component, maybe a solvent or maybe other solid. If it is solvent, it is called as solvent or the hydrates. And if it is multi-component system is there, that covers the co-crystals. So in the crystalline form, multi-component, as the name suggests, the co-crystals that consist of two components in one single crystal lattice structure. Next slide, please. So these are the two component co-crystals are the two component systems which are present. If you see, this is a diagrammatic representation of various crystalline forms which are available as API. The first represent the pure API molecule. Only single molecules are there. The B present the polymorphs. What is polymorphism? Anybody? Yes, those which are having the different arrangement, different physical properties into crystal lattice structure. Clear? That is a polymorphs of pure API. Then there may be hydrates or the solvents are there where solvent is associated, and then come the salt forms are there where there is a combinations of the two ions. That is salt forms are there, and there is last one cover the pharmaceutical co-crystals where the API molecule is combined with another moiety that is called as co-former. That is called as the co-former. Next slide, please. See, there are various problems which are associated with the API. The major problems are there: poor dissolution rate. Because you will know that maximum drugs which are available into the market, they are BCS class. Which drugs? M.Com students. Maximum drugs which are available in the market, they are of which BCS class? BCS class 2 drugs are there, which are at the low solubility and the high permeability. Yeah? So that's why you can see, because for permeation of the drug, the liquid lipophilicity is required. That's the maximum drugs are BCS class 2 drugs. So dissolution problems are there, solubility, moisture uptake, and major problem is the flow properties and compressibility of the drug, and along with that the stability of that particular drug. So these particular problems can be overcome by various approaches. That may be a complexation, that may be a salt formation, and one of the major approach is a crystal engineering or the co-crystallization or crystal habit modification. Because you know that one particular drug is available in various crystalline form, and out of all these forms, one form is more stable as compared to the other form. So that's why you can do the co-crystallization. You can go for crystal engineering, that is co-crystallization with crystal habit modification. Next slide, please. So crystal engineering, basically this is a very big term that involves the crystal habit modification as well as co-crystallization. The crystal engineering is considered to be designed and modify the growth of that particular crystal, control the growth of that particular crystal, so that you can get a crystals of desired physico-chemical properties. So crystalline, the growth, design and growth of crystalline molecule solids with aim of changing the material properties, that is called as crystal habit modification. So the crystal habit modification that can be done by various techniques, like changing the solvent systems. That may be changing the crystallization condition like temperature, rate of cooling, rate of heating, as well as that, then addition of sealing agent and addition of polymers. You might have done the practicals of medicinal chemistry. किए हैं? आपने recrystallization किया है? You are doing the recrystallization. During recrystallization, if you are suddenly cooling any particular solution, what happened? The crystals are small size. If you are keeping aside for overnight, you are getting the minerals and big crystals are there. So if you change the solvent, the size and shape of crystal become changed, and that will change the properties of that particular molecule, that particular API. So this is crystal engineering. It is one of the part of crystal engineering, that is a change the crystal habit of that particular drug. And next part comes is a co-crystals. Next slide, please. See here, many of you are doing your M pharmacy, maybe on the verge of selecting a topic for M pharmacy. Project work ke liye, right? Might be selected, or some after completion of B pharmacy, you are going to select one particular topic. And as far as possible, सबको लगता है कि मेरा topic novel होना चाहिए. It should be new topic, because the first question asked by the examiner: what is the novelty of your work? Here, what is the novelty of your? So this is a one particular topic. Co-crystallization is one topic you can take upon for the M.Pharm as well as PhD research work where you will get the novelty in that particular work. Okay? Because you are changing the properties of existing drug. You are changing the properties of existing drug. Next slide, please. It's a crystallization process. We will not go in detail. You might have studied the crystallization process during your UG level. आपने Myers theory of crystallization पढ़ा होगा? पढ़ा है कि नहीं पढ़ा? पढ़ा है ना? Short note लिखिए फिर 5 मार्क के लिए दिखाई देगा. I know because being a teacher, short note is a very popular question. और स्टूडेंट के लिए भी पॉपुलर होता है और टीचर के लिए भी पॉपुलर होता है क्योंकि क्या है कि कितना लिखना है वो स्टूडेंट डिसाइड करता है कितना शॉर्ट लिखना है कितना लॉन्ग लिखना है ठीक सो दैट्स व्हाई नेक्स्ट स्लाइड प्लीज द क्रिस्टलाइजेशन प्रोसेस नेक्स्ट term is a co-crystals. Now what are co-crystals? This is a recent concept which is introduced. Co-crystals are defined as actually complex of 2 or more neutral constituents. If you consider, in case of salt, it is not a neutral compound. In case of hydrated solvent, one is a solvent molecule. But here, this is a combination of 2 substances or 2 molecules which are neutral molecular constituents. They are bound together, very important, only by non-covalent interactions. Because what happens? Whenever there is a chemical interaction, there is a chemical change. And whenever there is chemical change, there is change in the properties of any particular drug. But if you are not chemically changing that particular properties, only your physical bonding is there with the one another substance. And if that particular complex is having the changed properties or required properties or altered properties as your requirement, that is advantageous. आप उसमें कोई chemical change कर रहे हैं क्या? No, because it's no covalent bonding. Only a hydrogen bonding is there or non-covalent bondings are there. Hydrogen bonding. Here, there is a formation of pharmaceutical co-crystals involve incorporation of API with another pharmaceutically acceptable molecule. Pharmaceutically acceptable means it should be a GRAS listed. What is GRAS listed? GRAS. M.Pharm students. सुना है नाम? Generally regarded as safe. That is called as GRAS. Okay? These GRAS listed drugs, this list is published time to time by the FDAs. Regulatory bodies are publishing this particular GRAS list time to time, and you have to select, you should select rather, whenever you are selecting any particular excipient, whenever you are selecting any particular substance for the interaction, it as far as possible, it should be selected from the GRAS list. It is generally regarded as safe list. So here pharmaceutically acceptable means it should be a GRAS listed, it should not have any toxic effects. Then the resulting multi-compound, this crystalline structure which is there, co-crystal, will maintain the intrinsic activity of the API. That is very important. Means there is a combination of co-former and the API, but the intrinsic activity of that particular API molecule is not going to change. So that's why it is very interesting field also for the pharmaceutical industry. Because what is happening, जैसे मैंने मेरा first slide ही था, the change in existing things. There are n number of drugs which are available in the market. अभी आपने देखा होगा, in every category, thousands of drugs are available in the market. And to invent one particular drug or to discover one particular drug, huge amount of money is required if you are developing from the scratch. If आपको you have to go for the moiety selection, then you have to go for the synthesis, then the preclinical, then clinical, all the phases. You have to pass all phase 1, phase 2, phase 3, then it will be marketed, and then post-marketing surveillance. That requires huge amount of money. So rather than that, you can change the properties of the API molecule which is already in existence. आपको मान लीजिए कोई drug है which is having the problem of solubility. The drug which has the problem of maybe of permeability, the problem of compressibility, problem of activity. Yeah? So that can be changed by these co-crystal techniques. You can combine that particular with a particular co-former so that you can tailor-made your API as per the requirement, as per the properties which are required. So that's why, and why industries they are attracted towards that, because the co-crystals there is an opportunity of intellectual property rights. Co-crystals can be patented. Co-crystals can be patented. You can patent that particular co-crystals, so that will increase the life cycle of that particular API molecule. You know that evergreening strategies of patents. मालूम है आपको? This evergreening strategy of into the patent system. What industries they are doing? Generally, what is the term of a patent? 20 years. So after 20 years, that what happened? That particular API will be out of patent. So what companies they are doing? They are developing different co-crystals, different forms, different polymorphs of that particular drug. That is called as evergreening strategy. Means there in that particular patent will be continued again for the next patent. Okay? So these co-crystals can be have the patent, IPR can be get on that particular co-crystals, and that will extend the life cycle of that particular API, and it will remain patented as a co-crystal. So that's why what is happening, the interest of the pharmaceutical industry is increasing toward this co-crystals. Next slide please. So, how many of you are heard about co-crystals? कोई वर्क कर रहा है co-crystals के ऊपर? Anybody? But I would suggest it's a very good area to work, even pharmaceutics students. And why this particular topic is selected? Because this is a combination of chemistry and pharmaceutics as well as pharmacology. Because whenever you are selecting that particular moiety, you should understand the chemistry of both the molecules. When you are going for changing the property, you should have the knowledge of pharmaceutics. And when you are checking the potency, you should have the knowledge of pharmacology. So it is an interdisciplinary type of research work that you can do. And that's why I selected this particular topic for this particular research, conference, because it's going with the theme of that particular conference. So here, these are the advantages of co-crystals already been discussed, because crystalline form, here there is no need to break the any particular bond. It is only a physical type of interaction. It is patentable, and most important, n number of co-formers are available. जो combination करना है. Numerous number of co-formers they are available into the list. Here you can select that particular co-former. You know that maximum drugs which are available, they are consist of certain groups which can form hydrogen bonding. Okay. So if any particular co-former, if you are selecting which is having the ability to form the hydrogen bonding, we'll see how that particular co-former is selected. So there are n number of co-formers are there. You can select the appropriate co-former. That's why these co-crystals can be developed for maximum type of API molecules. There high yield is there because yield is almost 100%. It is just a combination with almost all the methods except the spray drying method. Everywhere the yield is very high in case of co-crystal formation. Then green technology, अभी तो पूरा जमाना ही है eco-friendly. Green, yeah, green things is eco-friendly things are there. Yeah, so even you can go for flight tickets. हम फ्लाइट के अंदर बैठे होते हैं, तो भी हमको यदि ऐसा कोई फ्लाइट का टिकट चाहिए जिसके अंदर list it is more green or more eco-friendly, the costing is higher. Yeah, so that's why the world is going towards the green technology. Eco-friendly techniques are there. So that's why this you can use eco-friendly techniques like solar crystallization. You can use the microwave synthesis method. You can use the supercritical fluid technologies where it is considered to be eco-friendly techniques are there. Then it is attractive alternate for the drug development. हमने already discussed किया, because drug development is a very vast process. Rather than that, it is a very attractive alternative. Here, पूरा घर बांधना नहीं हमको उस तरह से. Here, अंदर का फर्नीचर ही अच्छा करना है. Here, घर को कलर अच्छा करना है, ऑटोमेटिकली नया लगने लग जाएगा. लोग बोलेंगे नया बन जाएगा. अभी तो हेड में दिखाते हैं, सिर्फ कलर दे रहे हैं, नया कंस्ट्रक्शन किया आपने. कहीं टीवी एड में देखते हैं ना, जैसे कोई एक एड आती है, नया घर नहीं, उससे कलर नहीं है, इसके पहले आती थी ना एड ऐसी, अभी भी आ रही है. Yes, so attractive alternate for the drug development and maximum crystallization component from the grass listed, so it will not have any toxic effects and it will not affect the pharmacological activity. Next slide please. थोड़ा कुछ समझ में आ रहा है? All of you? ये तो क्या होता है कॉन्फ्रेंस में, सबसे पहले तो ये होता है कि जितना लेट शुरू हो उतना अच्छा. फिर दूसरा एक होता है तालियां बजती है आखिरी में, तो सबसे ज्यादा जब सांस लेते हैं जब थैंक की स्लाइड आती है तुम्हें, और यदि बहुत जोर से तालियां बजी तो समझ लेना आई के लिए बजी. बट आई हैव अ पर्सन तो पॉजिटिव एटीट्यूड, तो मैं देखूंगा नहीं मेरे लिए बजी. तो आप किस रीजन के लिए बजाएंगे मुझे नहीं मालूम. सो ओके, इन केस ऑफ drug co-crystal, these is various characteristics are there. where अभी तक co-crystals का हमने देखा, एक co-former आप ले सकते, अभी तक पहले co-crystals जब invent हुए थे, that time the co-former is from various chemical categories. कोई acids लेते थे, amino acids were there, the carboxylic acids were there, amides were there, where there is the formation of the hydrogen bond. But later on, what happened? The concept was come into play. We just 2018 के बाद में. It's a very recent concept after 2018. What happened? Instead of one particular co-former, why not you should select another drug as a co-former? Here means आपको co-former जो select करना है, you are going to select another drug which may be having the similar pharmacological action or which will have the complementary pharmacological action. Complementary means required pharmacological action. Here, suppose you are selecting what is two NSAIDs. Here, so it will potentiate the activity. It will potentiate that particular activity. Here, if you are selecting the one antihypertensive, anti-diabetic drug, generally what happens? Antihypertensive, anti-diabetes, they are complementary associated diseases are there. अभी लोगों को दोनों होती है साथ में, होती है ना? Hypertension भी आता है और diabetes साथ में लेके आता है. So if you are selecting this type of combinations, rational combination if you are selecting, so you can change the properties at the same time, you can take the advantage of both the drugs simultaneously. So in another simple word, it is a complex but physical complex of two drugs. It is a physical complex of two drugs. So both the components in the co-crystals are the APIs. Out of that, one is a major component, जिसके आपको property change करना है. Other is a co-former, but that co-former is also from the API, that same pharmacologically active. It may be from the natural sources. You are taking natural sources because we are in the rich tradition of the natural medicines are there. Okay? So in that particular natural medicines, you can select one particular natural moiety as a co-former. Okay? And then you can potentiate the activity of the API molecule. There is a change in pharmacokinetic and pharmacodynamic of a drug. Both that will be changed. Pharmacokinetic is also changed, pharmacodynamic is also changed if you are developing drug and drug co-crystals. Here ADMA change हो जाएगा because resolution if you are increasing, obviously absorption and other parameters will be increased. If you are increasing the potential in the activity, pharmacodynamic will also change. Then it will overcome the problems of FDCs. जो अभी गवर्नमेंट ने कुछ FDCs बैन किए हैं, fix those combinations. So that can be overcome by these co-crystals. Delivery of dual drug therapy, two drugs can be given simultaneously. Then synergistic or additive effect is there. If you are giving the same category of drug, you can get synergistic effect and additive effect. What is synergistic? So additive और synergistic में क्या difference है? दो और दो चार is additive. दो और दो पांच is a synergistic effect. More than the addition. Okay? That is synergistic effect there. And life cycle management जैसे मैंने बताया आपको, if it is patented, you can increase the life cycle of that particular patent and you can improve the physicochemical property. Next slide please. See here, these are the basic three steps are there in the co-crystallization. The first stage is designing. So designing is that you are selecting the appropriate API molecule and appropriate co-former. Appropriate co-former and appropriate API molecule that will be selected. Basically, while selecting that, you should understand there should be a only sort of physical interaction like hydrogen bonding, van der Waals forces, or only a physical type of bonding is there. Then screening is you are developing the co-crystals by various methods. Here, the very commonly used methods are old methods like slurry conversion method is there, co-grinding methods are there, anti-solvent methods are there. But now newer and newer eco-friendly methods that will be introduced as microwave or maybe a solo crystallization with sonication, supercritical fluid extractions are there that can also be useful. And then the characterization of that particular co-crystals, we'll see which are the various methods for characterization. Next slide please. The most important step you consider for the co-crystal formation is a selection of co-former. That is most and prime critical important step. So selection of co-former is the most crucial step. because the total success story of this particular pharmaceutical co-crystals depends on the selection of co-former. Clear? So here, what is important for selection of the co-former? You should study the structure of API. आपके structure API का क्या है? And depending on that, you can select the appropriate co-former. Find out the intermolecular interaction with suitable co-former. Here, you can use various methods are there. So it should have the minimum hydrogen bonding or mineral interaction should be there with the co-former. You can use it should be a supramolecular synthetic approach is there. You should have the ability to form the synthons with the combinations. These synthons means the complexes. When the two same molecules they are combining with each other, they are called as homosynthons, and when the two different molecules are combining, that is called as heterosynthons. Yeah? Then generally graph crystals are there. Generally, as a thumb rule, your co-former should consist of these groups: carboxylate groups, amide groups, alcohol groups are there. They are particularly selected for the formation of co-crystals. So first step, as a thumb rule, primary selection करना है. First see कि उसके अंदर ये groups है क्या? COH group, amide group or OH group that is present here. Then use of chemical structural database is there. This is a paid database which is available where you can get the appropriate co-former for your API molecule. Chemical structural database, but it's a paid database which is available here. Then it is also found that those compounds which are showing the polymorphism, they are more suitable for co-crystallization techniques. So those compounds which are having more polymorphic effect or polymorphisms are there. And again, you should count the hydrogen donor and hydrogen acceptor groups in that particular API molecule. Yeah? And then you can select the API molecule as another API, co-former as another API for the pharmaceutical drug co-crystals. Next slide, please. There are various approaches are there for selection of co-formers. Just brush it in short. First selection of co-former, hydrogen bonding आपको देखना है. For that you can use the graph set notation system is available. Here graph set notation system is there, which is a software where you can study the pattern of hydrogen bonding from the graph analysis. From the graph analysis, this is a system you can use where basically what is hydrogen bonding? Anybody? What is hydrogen bonding? हम कई बार यूज करते हैं हाइड्रोजन बॉन्ड. कौन बोल सकता है? हम फर्स्ट ईयर से पढ़ते आ रहे हैं हाइड्रोजन बॉन्ड. यहाँ देखो कुछ लिखा है, यहाँ दिख रहा है आपको. यहाँ कौन सा हाइड्रोजन बॉन्ड है? ये हाइड्रोजन बॉन्ड है, ये हाइड्रोजन बॉन्ड. डैश, राइट. तो ये डाइजेस्ट क्यों दिया? It is just an electrostatic attraction. Just an electrostatic attraction. Here, what hydrogen bond? When the hydrogen atom is attached to two electronegative atoms, one with a covalent bond and another with a electrostatic attraction. Electronegative atom चाहिए उसके. When a hydrogen atom is attached with two atoms, two electronegative atoms, one with a covalent bond and other with a electrostatic attraction, that is called as the hydrogen bonding, and we are more interested in that particular electrostatic attraction. Next slide please. So generally the 6-member ring is present and is more preferred in the structure for the hydrogen bond formation. Next slide. Then another thumb rule is difference in pK. pK आपको मालूम है pK की स्टैंडर्ड वैल्यूज होती है. API का pK होता है और co-former का pK होता है. So as a thumb rule, what you can select if the pK difference between the co-former and the drug is less than 3, 0 to 3 के अंदर है, the formation of co-crystal is possible. More possibility of formation of the co-crystals. So second method you can use a pK difference method is there for selection of the co-former. Third is a very calculative method that is called as a very good method, Hansen solubility parameters. Solubility parameters are the contribution. You can see the contributions of the different groups into the solubility, that is in terms of solubility parameters. So here the total cohesive energy of that particular drug is calculated or API is calculated and the co-formula is calculated by splitting that particular molecule into the different federal group contribution method. In the federal group, and the energy values, the energy values they are available, and from this particular formula you can calculate the Hansen solubility parameters of the drug and the co-formers. Next slide please. Not go in detail here because the software is also available. Hansen solubility parameter का software भी available है. You can put your drug, you can get the Hansen solubility parameter calculations of your drug, then Hansen solubility parameters of the co-former. Okay? And then the difference in that Hansen solubility parameter of the drug and the co-former is less than 7. It is the chances of co-crystal formation is more. They are considered to be miscible and the chances of co-crystal formation is more. If the difference in the Hansen solubility parameter of the drug and the co-former is more than 7, then they are considered to be immiscible and there is a least chances of formation of the co-crystals. Next slide please. Then another technique in silico technique. In silico technique is that in silico technique is a different molecular docking software you can use. You can use docking softwares are there and use like docking software you can calculate docking scores and the energy score you can calculate and you can predict the co-crystallization. क्या हो सकता है? See, you can use the different, these are the Autodoc and other softwares which are available, light software is there, and you can study the interaction energy, free energy binding, hydrogen bond, van der Waals interaction, hydrophobic interaction, pi-cation interaction, bond distance, molecular conformation, all that can be calculated by using the softwares. आपने molecular software तो बहुत सीखा हुआ, Federer है, Like है, Glide है, different softwares है. We use that particular software in our co-crystallization. So you can use this in silico technique for prediction of the co-crystallization. Next slide please. Then next method is also which is there. It's called as the saturation solubility techniques. आपने कभी किया होगा cyclodextrin के ऊपर किसने काम किया? Solubility enhancement cyclodextrin very नहीं किया clear. So here in case of cyclodextrin, what you are doing, you are calculating the increase in solubility in presence of cyclodextrin. So here what is done in case of saturation solubility study, the solubility is measured, saturation solubility of API that will be measured here and co-former is measured separately, and then the API and co-former they are mixed in the formula system, and again the solubility is, the temperature is increased slowly with 0.3 degrees per minute. Here the heat will increase and what will happen at one particular stage the solution become clear. Solution become clear, that is called a saturation stage, and if that saturation temperature is more than the 10 degrees as compared to the reference temperature, means for solubility of API, then you can consider that there is a formation of co-crystals. And crystal cocktail method is there. It is a very crude method. Clear? जैसे cocktail का नाम आ गया तो बिल्कुल clear. So किसी किसी की नींद उड़ गई, किसको लगा ये हाँ ये अच्छा याद आ गया. So co-crystal cocktail method is that. Here what is done in this method, more than 3 co-formers and they are co-grind with the drug. Here 3 से ज़्यादा co-former दे लिए, so co-grind किए और उसका IR निकाला. Here and then with the drug and heating. So what will happen? There is a formation of hydrogen bonding after grinding. After grinding on a crude method, and then they are analyzed by IR and thermal techniques. Thermal techniques or IR से उसको analyze किया, you will find certain hydrogen sort of hydrogen bonding will be there, and there will be change in the thermal activity of that particular API molecule in presence of this particular co-former, and then accordingly you can select. It's a crude method. पहले 3-4 की बात कर लिए, इसमें से एक select किया, फिर Hansen solubility parameters लगाए. So accordingly you can filter it out. Here, सबसे पहले आपने हाइड्रोजन बॉन्ड के हिसाब से देखा, then you apply Hansen solubility parameter, then you apply API, maybe in silico technique, PK technique, and you are filtering out, and ultimately you will come to conclusion that these 3 or 4 co-formers are there which can form the better co-crystals with this particular drug. Next slide please. So selection of co-former is there, which can be done here. Then there are different methods for co-crystallization. We'll not go into just the names are there. So traditional techniques are the solvent evaporation technique, then solid state mechanical milling or grinding is there, where the co-crystal can be formed, or it may be a solvent induced technique, slurring techniques, then solvent drop technology. In that, just simply a drug and a co-former and API is taken, it is grinded in presence of solvent, it is milled in presence of solvent, that will lead to formation of co-crystal. These are the traditional methods which are available in the literature. But now advanced techniques are there, which are there microwave assisted technique. सभी कॉलेज में microwave होगा. And see what is the beauty of this particular topic. The instrument requires, when if you see, they are very less. Even pacer and motor आपके पास है, APIIR co-former है, you can go for co-crystallization. बाकी तो आपको जो analysis करना है, that can be done outside. In IR सभी के पास होता है, सभी कॉलेज में IR है. So from IR you can predict the hydrogen bonding, उतना primary है. Then for XRT, DSC, can if you are having the in-house facility, then it's okay. Otherwise हम हमेशा क्या करते हैं, sampling we are outsourcing. So this is a very simple topic. हम ये नहीं बोल सकते कि सर इंस्ट्रूमेंट अवेलेबल नहीं है, क्या करें, फैसिलिटी अवेलेबल नहीं है, क्या करें, कहाँ से नॉवेल्टी लाए. और ये भी होता है सर नया ड्रग नहीं, यहाँ पुराना ही ड्रग लग रहा है. कई बार हम वाइवा के लिए जाते हैं तो ये ड्रग क्यों सिलेक्ट किया, तो वेरी क्लासिकल आंसर फ्रॉम द स्टूडेंट, सर यही अवेलेबल था. एक गाइड ने दिया, दो गाइड ने दिया, गाइड भी क्या करेगा. प्रॉब्लम इज दैट n number of M.Com students are there, pharma industry limited है, और हम सैंपल्स कितना मांगेंगे. How many samples we can ask to pharma industries, limitations are there. So that's why you should go for this type of topics where you can use the traditional APIs are there. आप नया API नहीं मांगोगे इंडस्ट्री से, इसका 1 मिलीग्राम का कीमत कई हजार में है. कई बार होता है ना बस कि एंटी कैंसर ड्रग मांगते हैं और स्टूडेंट्स उनको 100 ग्राम 100 ग्राम ऑफ दिस पैजोपिरिक भेजो, वो बोलता है को कॉस्टिंग मालूम कितना है 100 ग्राम का, और आप एज ए गाइड हम भी साइन कर देते हैं. हमारा भी क्या है, नहीं 100 ग्राम लिखेंगे तो 10 ग्राम तो भेजेंगे. हम वो भाव करने वाले लोग हैं. 100 का बोलो तो हम 10 में मांगेंगे. तो वही हमारे लॉजिक यहाँ लगाते हैं. तो जैसे आप 100 ग्राम का रखते हो बाजू में रख देते हैं, शायद इसको कुछ मालूम ही नहीं है, ही इज आस्किंग 100 ग्राम ऑफ दिस सैम. अभी मेरे पास है ल्यूलिकोनाजोल, वेरी कॉस्टली ड्रग, 100 ग्राम ऑफ ल्यूलिकोनाजोल, सर इतने मंगाना है. 100 ग्राम ल्यूलिकोनाजोल कितने का. So that's why you can go for this type of topics where traditional equipments है, traditional आपके जो भी API molecules है, and co-formers are there, then you can select and you can have the good novel findings. So these are the techniques are there. Ultrasound assisted techniques, ultrasonics बहुत जगह है, maximum colleges में available ultrasound techniques are there. Then spray drying is also available. In many colleges spray drying is available. Then even high shear granulation, these are the again industry technique. Freeze drying भी बहुत जगह available. Life lasers are there, available freeze drying techniques are there. Then laser irradiation, field screw extrusions are there, electrochemical induced and resonance acoustic mixing. These are the other rare techniques are there which can be useful for the formation of co-crystals. So you can go for advanced technique also in maximum colleges if you find. And what you can do, you can select the different co-crystals and different methods. What you can go for your research topic, what I am talking about. Then you can select one co-former, different methods, different co-formers, one method that can be selected, and you can come out with a very beautiful finding that this particular method with this particular co-former is giving the good results. Okay? So that sort of work you can do into the even chemistry, pharmaceutical quality assurance department. Next step please. But more important, students के लिए ये भी होता है कि हमें वर्क क्या करना है. कई बार होता है कि when you work in pharmacy, गाइड्स बोलते जाओ तुम्हारा इंटरेस्ट का क्या है देख के लो. आफ्टर कंप्लीशन ऑफ बी फार्मेसी उसको ये नहीं मालूम होता जाना कहाँ है, क्लियर, एंड व्हाट इज द इंटरेस्ट. सो दैट्स व्हाई दीस आर द वेरियस थिंग्स यू कैन डू. देयर इज द कैरेक्टराइजेशंस ऑफ दिस पर्टिकुलर co-crystals. A very simple characterization, सबसे पहले melting point. You know that whenever there is any physical change, there is change in melting point. Melting point change हुआ, इसका मतलब क्या हुआ, there is certain physical changes are there. Because melting point, as melting point reduces, solubility increases. So आपको melting point से पता लग जाएगा कि whether solubility increase हुई है या नहीं. थोड़ा सा melting point कम हुआ, इसका मतलब थोड़ी सी solubility बड़ी होगी. नहीं तो हमारे बड़ा के ही लाते स्टूडेंट है ना. Because see in the हिस्ट्री ऑफ ऑलमोस्ट मोर देन 14 years ऑफ फार्मेसी, मैं हमेशा बोलता हूँ किसी स्टूडेंट की बैच फेल नहीं हुई, स्टेबिलिटी हर एक का रिलीज 99 आया हुआ, किसी स्टूडेंट के पास अनस्टेबल तो बनी नहीं। इंडस्ट्री इज वर्किंग फॉर 4 years 5 years, हमारे स्टूडेंट इतने ब्रिलियंट है 2 महीने में पूरा स्टेबल बैच विद रिक्वायर्ड रिलीज, रिक्वायर्ड एनिमल एक्टिविटी, है ना, क्योंकि एनिमल एक्टिविटी में आई जाती है, एनिमल को क्या मालूम बेटा को क्या करना है, वो कुछ उठाई किसने देखी, है ना, so that happens, so that should not happen in the research here, whatever, even there is a journal of negative results, आप देखिए journals of pharmaceutical negative research, so in negative result में that is also a positive impact that can also be published, लेकिन कई बार आपको ये होता है कि हमेशा पॉजिटिव ही आना चाहिए, that happens in the research here, so these are the melting point, stability at various conditions, you can check stability chamber सभी के पास है, here you can keep that at various stability chamber positions, here as per the ICH guidelines you can check the stabilities, then you can change the solubility because your main objective you can go for saturation solubility studies, then you can go for intrinsic dissolution rate, IDR, what is IDR? Right? Because you know that the solubility of any particular drug is changes with surface area. But if you keep surface area constant, then what is the solubility, what is the dissolution of that particular drug? That is called the intrinsic dissolution rate. If you see the type 7 apparatus of dissolution, disc, मालूम है ना? 7 टाइप के apparatus मालूम है? उसके अंदर एक disc apparatus सबसे 7 वाला. So in that one particular, you are converting that APIIR molecular co-crystals into a non-disintegrating disc, very hard disc with maybe a higher press, and then you are putting so that only one surface will be exposed to the solvent, and the dissolution is carried out, and the unit of intrinsic dissolution rate is milligram per centimeter square per hour. Means what is the solubility of that particular drug per unit area per unit time? That is called as intrinsic dissolution rate. That is a very good parameter that you can calculate. ये आपके पास हो सकता है, हमारा टाइप 7 apparatus नहीं है, किसी के पास है, किसी के पास है नहीं है, टाइप 7 किसी के पास नहीं है, ये टाइप 7 apparatus जनरली नहीं होता है, कॉलेजेस में भी होता है, वहां टाइप 2 हो तो बहुत हो गया, क्लियर, सो टाइप 7 तक हम जाती ही नहीं है, बट व्हाट यू कैन डू द मॉडिफिकेशंस, हमने क्या किया हमारे पास कि मेन एम है कि एक सरफेस एरिया एक्सपोज होना चाहिए, तो कैल्पोल की टैबलेट का एक रुपए की टैबलेट लेके आ गई, उसका एम्प्टी शेल निकाला, हमने उसी शेप का एक डिस्क बनाया हमारे ड्रग का, हमारे को क्रिस्टल्स का, देन फिटेड इनटू दैट, उसको फिट किया और उसको क्या किया, बास्केट के नीचे 2 साइडेड टेप से चिपकाया, समझ में आया कि हमारा मेन एम क्या है, ओनली वन सरफेस एरिया एक्सपोज होना चाहिए, एंड वी कैलकुलेट इंट्रिंसिक डिसोल्यूशन रेट, एंड दैट इज एक्सेप्टेड बाय द पेपर्स because of principle same है, principle similar यूज किया और apparatus लिखा है, we modified the condition because for informed research work आपको principle मालूम होना चाहिए why you have done this, so you can tell intrinsic dissolution rate, XRD studies are there, then very good method is single crystal XRDs are there, but unfortunately बहुत कम जगह facility available single crystal XRD की, you have to separate that particular crystals, a single crystal आपको separate करना पड़ेगा, and then you have to calculate do the XRD studies, infrared studies, Raman spectroscopy, DSC is another very good tool that is available at many places. DSC maximum jagah available hai, even you are going for outsourcing, the cost is not too much. Raman spectroscopy की भी cost कुछ बहुत ज़्यादा नहीं है, that is just for your information. All these IITs, sophisticated instrumentation sections, अपने सैंपल जब भेजोगे सभी IITs के that is called as SAF, ये SAF अली खान नहीं है, SAF sophisticated instrumentation sections are there, you can send the samples, and where the very economic rate of this Raman spectroscopy and the DSCs are there, that is affordable one. Then the NMR techniques, then you can carry out scanning electron microscopy, SEMs are there, then polarized light microscopy is not it is available at many colleges, you can change the crystal habit by this particular polarized light microscopy, hostage microscopy and gravimetric analysis are there, thermogravimetric analysis, PGA techniques are there. These are the various techniques you can use for the analysis of the co-crystal formation. Next slide please. So it is not necessary to carry out all the analysis. आप इसमें से कुछ representative कर सकते हैं. And if you want that your paper, research paper should be accepted at very high impact journal, accordingly you can go for the analysis. These are some examples of drug co-crystals which are available under this, which are into the study research work. Drug tramadol, celecoxib, aceclofenac, paracetamol, or meloxicam with aspirin. So these type of, you can see these are complementary with each other. Okay? So for NSAIDs, anti-TB, so maximum work which is done into the drug co-crystal is anti-TB. Why? Any reason? Because anti-TB में single dose therapy generally fails after some time. Resistance is developed. So here you can combine two anti-TB drugs simultaneously with the different mechanisms. दो अलग-अलग mechanism के drug you can combine with each other, clear? And that can give a dual effect. So resistance into the anti-TB therapy that can be reduced. So that's how you find maximum research जितना भी है. And we also at our college we have done this particular research on anti-TB drugs. Then Bidaquilin. Okay, next slide please. So you can see various examples. Even in market also commercially, very few drug co-crystals are available. But it is commercially available means this concept is accepted. ये commercially available हो गया. इसका मतलब क्या है? This is accepted by the FDAs. So example is this Interdisciplinary is a drug which is a combination of valsartan sodium and sacubitril sodium for heart failure. Then the dimethyl that is dimethyl and the 8-chlorotheophylline for motion sickness. So this is a combination. These are the two co-crystals, drug co-crystals which are available into the market commercially. So commercial acceptance is there. Then the challenges in development of drug co-crystal. First, no guarantee that synthesized drug co-crystal is pharmaceutically acceptable combination. Because what happened? One of the research paper I find there is a combination of theophylline and phenobarbital. Drug co-crystal developed theophylline and the phenobarbital. Theophylline is a stimulant, phenobarbital is a depressant. Then what is the logic in this particular drug? Drug co-crystal ban jayega, but there is no logic. So while selecting that particular moieties for the drug co-crystal, the therapy that should be taken into consideration and both the components should be complementary with each other. So it is not practical rational. There should be practical rational. Again, the co-crystals have at least fixed stoichiometric ratio. Co-crystals ऐसा नहीं कोई भी combination से बनता है. While preparation of co-crystals ऐसा नहीं कि आपने just किया और बन गया. Different stoichiometric ratios that have to be tried. At one particular stoichiometric ratio, you are going to get a good co-crystals. So fixed stoichiometric ratios are there, which are different from the oral dose. हो सकता है उनका oral dose different वाला दोनों का भी, लेकिन इसी ratio पे वो बन रहे हैं. Suppose the dose of paracetamol is 500 milligram and aceclofenac is 100 milligram. But in co-crystals, जहाँ पे ऐसा हो रहा है combination, when you are taking the one part of paracetamol and two parts of aceclofenac, तभी co-crystals बन रहे हैं. So no risk. Okay. So this is a challenges which are there. Okay. So there is no, what are the ratios which are required that may not be matching with the actual dose of the drug. And then selection of appropriate combination is very challenging. And again regulatory ambiguities are there. Because पहले तो pharmaceutical co-crystals का acceptance पहले नहीं था. अभी वो आया है. Again drug co-crystals का acceptance है, that is still a question mark at many regulatory bodies. Okay. Next slide please. So regulatory ambiguities are there. But important is APIIR. You are getting the APIIR into the pharmaceutical co-crystal because the three characteristics which are required for the APIIR. कौन सी तीन चीज लगती है? पेटेंट के लिए क्या चीज लगती है? तीन. कोई भी रिसर्च को पेटेंट करने के लिए कौन सी तीन चीजें लगती है? सामने देखो. Novelty, non-obviousness and utility. These three things are required. Novelty is required, non-obviousness is required, and there is another and third is a utility or industrial applicability that is required. So novelty तो है, because आपका old drug है, you are doing a new combinations you are creating, so obviously novelty is created because many co-formers are available and you can select a novel co-former and that combination becomes novel. So novelty is not a questionable. Obviousness is there because n number of co-formers are there, n number of techniques are available for proving the co-crystals formation. So non-obviousness will be overcome, and utility, commercial advantage are much more. Already हमने discuss किया. So ये तीनों भी जो characteristics which are required for patenting of any particular research work that is qualified for the pharmaceutical co-crystals, and that's why now pharmaceutical co-crystals are patentable. You can have the patent on the pharmaceutical co-crystals. Next slide please. इसके पहले नहीं था, but since last 3 to 4 years these co-crystals are patentable. The regulatory aspects, again there is even US FDA and this European medical agencies are there. They are having the ambiguities into the definitions of the co-crystals. Definition of the co-crystals. Here. So here, likewise, this US FDA said that these are the crystalline material composed of 2 or more molecules in the same crystal lattice structure, and European medical agencies say that homogeneous crystalline structure made up of 2 or more components in a definite stoichiometric ratio where arrangement in the crystal lattice is not based on the ionic bond. Here it is, but EMA except, or you can say they are compare the co-crystals and salt. Salt और co-crystal उन्होंने same category में रखे in case of EMA guidelines. Here, next slide please. So these are the case studies what we have done at our college. So this is a recent patent that we published. This is work done by one of my MPharm student, and also she got an award as the best thesis award at national level at Pune. They have everywhere. And more important than she is a graduation from Nagpur. Saikheda Sonikar College. Sonikar is very small college I think. But I will Raisoni college. वहाँ से थी वो graduate की, and later on we come for post graduation in our college. So for this project work, also got the best thesis award for this. So this is recently patent is published Indian patent. So drug co-crystal of bidaquiline. We developed bidaquiline co-crystals with the different co-formers, naturally occurring co-former. We have also done the anti-TB activity in vitro, and we found that the anti-TB of bidaquiline is increased as compared to plain bidaquiline. Okay. And many positive things that happen. That's why this is published. Then next, we also got a UK patent for this design patent because we developed a new method for the development of co-crystals. Okay? So we get a design patent for this new co-crystallization method. So we get a design patent for this particular technique. Next slide, please. So these co-crystals, you can drug co-crystal is recent. So this is our paper that is solubility enhancement of carbon using drug co-crystallization. So it is a recent technique. So this is a paper of somebody else, but and you can see one thing is that while going through literature, the maximum papers are from Indian authors. Here drug co-crystal से maximum paper है from Manipal University. Manipal University से बहुत ज़्यादा papers है इसके अंदर, and after 2018, 2019 के पास है. Rather अभी review articles आए कुछ 2020, 21 और 23 में. When you are observing a review article in a good journals recently, that means the recent topic. अभी अच्छे जर्नल से review articles आ रहे हैं, इसका मतलब क्या है? It is a topic is under development. Okay? So you can see this is a drug co-crystals Asian Journal of Pharmaceutical Science 2021 का article है, hypotheses and challenges. So these are the review articles are there, which is recently been coming up. So which show that there is a lot of potential in this particular topic if you develop. This is again recent in crystal research technology, the review of 2023. Drug co-crystallization promising approach to overcome barriers in pain management. So specifically, they have carried out the research onto the drug co-crystallization in the analgesic and antipyretic in pain management. And again, this is recent article in crystals with a very good impact factor. You can see maximums are there. Combre, Mahalakshmi Radhanan, these all are Indian authors are there. They are doing this particular research. So this is specifically onto the anti-cancer drugs co-crystals, drug co-crystals. So you can see anti-cancer category, anti-TB category, and the pain management. These are the areas where this can work very good. Next slide, please. And very important slide. Concluding remark. So, 1 hour was allotted to me. So, concluding remarks. So, co-crystal is a promising approach to improve the physical properties of API molecule. We already discussed a very good approach which is available. Again, this drug co-crystal is totally a new concept which can change the API industry. Because API industry को बहुत ज़्यादा even Rolling Kurana is there. मेरे senior थे. He is working on another one of my favorite topic co-process excipient पे काम कर रहे हैं. Excipient तो बहुत सारे हैं industry. But he is working on, and my recent topic is the co-process excipient. Co ज़्यादा work करता हूँ. Co-process, co-crystals, because there are novelties there. So these are the areas which are coming up. So this is my urge to the M.Pharm students and the molecular researchers or B.Pharm जब आप जाओगे भी M.Pharm के लिए, you should select this type of topic which is real applicable to the pharma industry. मैं ये नहीं बोलता कि आप तो क्या होता है कि सर ठीक है ड्रग है माउथ डिसॉल्विंग टैबलेट बनाता माउथ डिसॉल्विंग फिल्म बनाता clear but rather than that you can go for this type of topic of co-crystallization co-process excipient आप लोगों ने नाम सुना होगा रोलिंग खुराना का मैक्सिमम लोगों ने सुना होगा because a very big name in the pharma industry excipient industry specifically yeah so it's from the department हाँ सर डिपार्टमेंट मेरे सीनियर ही थे वो मार्गोनस पर नीतिका फार्मास्यूटिकल्स आईपीसी में आए होंगे ना आप लोग हाँ नीतिका फार्मास्यूटिकल हाँ बहुत याद आ रही नीतिका हर जगह पे नीतिका लगा था ना दैट इज रोलिंग खुराना एंड द प्रेजेंस इज अक्रॉस ग्लोब yes across Smiley across globe so he is working on co-processing of excipients so change of excipient तो बहुत सारे available market में लेकिन you can change the properties of excipient by using various methods. So similar topic is this one also, where it can change the face of API industry, where the old APIs can be also changed with the properties. But again, there is a need for development of robust model for predicting drug formation, and there are many challenges are there. Still, a journey is ahead. ऐसा नहीं कि आपको ये बस drug को पुस्तक हो गया immediately हो जाएगा. This is just a nascent area now. But it will play an important role in the pharmaceutical industry. It will take a long journey. Definitely, lots of challenges are there. Many challenges are there. The challenges includes the selection of co-formant. Challenges includes regulatory hurdles. सबसे बड़ा hurdle तो हमारा regulatory hurdles are there. As far as industry point of view is concerned, so regulatory hurdles are there. But still, it is attracting the pharma industry because of the only one concept, patentability. It can be patented, so it can increase the life cycle of that particular drug. So that's why it is attracting the pharmaceutical industry towards this drug co-crystals. Next slide, please. So definitely, I think you are going to get some idea of pharmaceutical co-crystals, co-crystallization process, pharmaceutical co-crystals. So you can take upon this type of topics, even for your PhD research work. M.Pharm का हो गया. PhD में सबसे पहले आपको novelty चाहिए. So you can do the novel and you can work in this particular topic जब तक आपको चाहिए. गाइड ने कितना वर्क करना है, कितने को फॉर्मर सिलेक्ट करना है, उतने सिलेक्ट करते रहो, मेथड सिलेक्ट करते रहो, and you can publish a different papers on one method itself, the different co-formers. So thank you very much for your patience listening. A road of success for you. I wish you all the best. All the success always. So thank you management of Raisoni group of institutions for giving me opportunity to interact with such a patient listening audience. Really good, because to get a good audience is also requires luck. And I'm lucky speaker that I get your the audience of your time. So thank you very much. Any questions are there? Try to answer or we can discuss that only. Yes, pharmacognosy, because recently जैसे मैंने बताया आपको कि for anti-TB drug we have selected co-formula from natural category. बहुत सारे natural anti-TBs है. तो anti-TB drug से we extracted the certain components of anti-TB and that component extracted then we couple with the anti-TB drug. So it is a combination. आप natural sources से कोई co-formula ले लो, co-formula natural source से लो, उसको couple करके देखो. ऐसा नहीं कि एक ही अपने लिया हो सकता है कपल क्लियर आपको फिर से उसका पूरा स्ट्रक्चर निकालना पड़ेगा यू हैव टू कैरी आउट ऑल दिस स्टडीज क्लियर नहीं Yes, that will require long study. That will require study. But once एक यदि आपका successful हो गया, because idea का base नहीं है, बड़ा idea डूब गया लेकिन base नहीं अच्छी है. One idea can change your life. Clear? So the one idea can change the scenario definitely. है ना? So what you say that we have done that एक entity filter को couple कर सकते. ऐसा कोई भी because we are in the vast era. अपने पास पूरा है, आयुर्वेदा में पूरा है. पूरे नेचुरल सोर्सेस के बहुत सारे ड्रग्स हैं, लेकिन आपको उसमें सेपरेट करना पड़ेगा कंपोनेंट एंड देन यू हैव टू कपल. यू कैन नॉट कपल टोटली अ क्रूड एक्सट्रैक्ट्स. थैंक यू.
Any questions from sir?
If there is no question,
Yes.
सर मेथड डेवलपमेंट में हम को रिसर्च का अप्लाई कर सकते हैं क्या इन केमिस्ट्री?
in analytical method development. Analytical method, see what is the objective of co-crystal basically is enhancement of the physical properties of the drug. So I don't, because our aim is to enhance the physical properties or to enhance the PK or the pharmacodynamic pharmacokinetic of the drug. For method development you can use other complexity, जैसे कि you are in the mixed solvency approach जिसको बोलते हैं. Mixed solvency approach है like you can use that mixed solvency. You can just refer the papers of Dr. R.K. Maheshwari from Indore. You know, Dr. R.K. Maheshwari is doing lots of work on this mixed solvency approach for method development. Yeah? So he developed a method for analysis without solvents with mixed solvency approach. Yeah? So that can be done. But I think co-crystals का case I reported it because our objective is totally modification of properties of that chemical. Rather, I would say he will make it more complicated because हमको interference कम चाहिए. Interference कम चाहिए. If you are adding additional component, again the interference will be increased.
Any other questions from sir?
Thank you very much.
Thank you, sir, for joining us today and adding value to our knowledge.
I would like to request respected Dr. D.C. Sahu sir, Dean School of Pharmacy, Raisoni University, Saikheda, to present a memento and to serve. Moving further, I would like to hand over the stage to my colleague Sakshi Thakre and Vanshika Lalwani for further sessions. Thank you.
Thank you so much, Vaibhav and Nikita. So now, moving forward, let's begin our next case session.
Our next case session is going to present by Dr. Jayaprakash N. Shetty sir. Sir is presently working as Associate Professor at Y.B. Chauhan College of Pharmacy, Aurangabad, India. He has completed his Masters from Mumbai University and completed his doctorate degree from Dr. B.A.M. University, Aurangabad. Sir has around 21 years of professional experience and also including in top 10 researchers list published by researcher of Stanford University. Sir has 6 patents to his credit and has published 261 research papers in international with a cumulative impact factor more than 600. He has received Best Paper Award in 2015 and also been awarded with Young Pharmacy Teacher Award. Now, we feel extremely honored to have you here, sir.
Now please welcome Dr. Jayaprakash N. Shetty sir with a huge round of applause.
So, very good afternoon. So, after such an informative lecture, which has been delivered by Dr. Lawrence, my teacher. So, practically it is difficult for me to continue with that particular motto. And we are started with the morning session. Now, basically, at the start, I would like to acknowledge the organizers of this international conference, and the theme of the conference is interdisciplinary research. As far as India is concerned, we are very lagging behind as far as interdisciplinary research is concerned, because in last almost 60-65 years, we could give only 2 molecules to the world, and the reason behind very few molecules which we invented or which we have given to the world. The reason is lack of interdisciplinary research, and this particular aspect needs to be explored. And if you explore this particular aspect, because we have tremendous mind, but we people are not ready to sit together. Pharmacology person is maintaining his ego. He is only concerned with the development pharmacological aspects. Synthetic chemistry person is always concerned with the synthesis of drugs, whereas toxicology is always concerned with the toxicology. But we are not able to sit together, and that's why we are not able to give many more molecules, and that's the reason. And that's why this particular theme is very important. Rather than, I can say that organizing such conferences, which are helpful for involvement of all sections of the science, and will lead to more, we can say, output as far as Indian pharma is concerned. Now, with that view, I'm here to deliver my session on this particular topic that is based on upcoming ICH guideline. Now, yes. Yeah, why I'm saying this upcoming ICH guideline, because what is ICH? That is most important. There are 2 important terms that I want to highlight specifically: ICH and quality by design. Now, what is ICH? Most of you are B.Pharm students and M.Pharm students. What is it? So this is nothing but a particular council that helps, particularly brings regulators and manufacturers together in order to bring the harmonization as far as the technical requirements in the pharmaceutical sector. And this particular aspect has been taken into consideration by the regulatory bodies because of certain issues that has been faced by the regulatory bodies in the past years. Since we are working on the different drugs in the different parts of the world, but regulators are different, and that particular regulators, they were regulating the pharmaceutical activity in the different part of the world, and there was no harmonization among them. And to create that particular harmonization, this particular council is working, and this council regulates the quality of the pharmaceuticals and different activities of the pharmaceuticals, the different guidelines. Now, different guidelines which has been given, the different amendments that have been implemented from time to time, they are controlling the different pharmaceutical activities that are, we can say, working with the common objective, that is quality products. Next slide, please. Now, before going to this particular session, as I mentioned, there are 3 important terms that we need to go through: ICH and quality by design. Now, many of us are going about this quality by design because we are talking about this particular term almost, say, 1 decade. So this is not a new term. This is the term which we are using across the science, specifically the engineering science, almost 60 years back. This particular science, it is well explored by engineering science. This particular approach has been well used, well documented by the engineering science, specifically chemical engineering science, where they have solved many issues associated with the process of any of the, we can say, the chemical preparation or identification. Now, what makes pharma industry to accept this particular science as far as the pharmaceutical development is concerned. We are accepting something new only because we are facing the problems associated with the existing concept. If a new concept has been introduced in the market, if the new concept has been introduced or accepted by the regulators, the reason behind acceptance of this particular new concept, it is only because they want to address certain issues that the regulators or the manufacturers they are facing. Now, as such, quality by design, it is a systematic approach. Basically, it works on predefined objectives. So, the current scenario which we are following in the pharma sector, it is quality by testing. We are taking the decision about the batch of the tablet or batch of a particular formulation by performing the testing on a small sample size, and we are taking the decision about the entire batch. Now, if that particular sample size, if it is not sufficient, if that particular batch gets failed, there will be economic loss as far as the pharmaceutical development process is concerned. Now, there must be a need to overcome number of issues associated with the pharmaceutical industry. So, as I mentioned, this is not a new science. This particular science has been well accepted by the engineering field. If you see the research papers published in 1960s, we can see quality by design approach in the chemicals engineering field. So, this particular science we can say accepted in 2000 almost 4, not directly, but through implementation of different Asian guidelines. So, this quality by design, it identifies the critical parameters which are affecting the performance of the method. If you are working on the product, it identifies the properties of the product that are critically affecting the quality of that particular product. So, quality by design, it is a concept which we can apply to any process, which we can apply to any product also. Next slide, please. So, this is just a difference. So, the very basic objective of this particular presentation is to make aware about the concept of quality by design and how ICH is slowly implementing basic concepts of quality by design to the different guidelines. So, these particular guidelines, they are slowly getting the control on the pharmaceutical regulations, and on one fine day, by the time you enter in the market, you are B.Pharm finalist students, you are M.Pharm students. If you enter in the market, and if you want to go on analytical method development, then you must have a clear idea about what this ICH 14 is. Now it is the primary stage. This concept paper has been published in 2018 as far as ICH 14 is concerned, where implementation of this analytical quality by design, what quality by design and what analytical quality by design, we'll see in the latter part of this particular presentation. But before that, what is quality by design? It is a science and risk-based approach. It is having some principle of understanding the process. So, if you want to gain the control of any process, the basic fact behind this quality by design is, if you want to gain the control of any process, then you must understand that particular process. If you want to success in any of the process or any of the step in your life also, then you must have a proper control on that particular process. So, gaining control, it is the most important aspect as far as quality by design is concerned. So, for gaining the control, you should know what that exactly process is. Which are the parameters involved? Which are the factors that affect the performance of that particular process? So, what is the objective of that particular process? To achieve that objective, which parameters may affect that particular process? And gaining the control on that particular aspects, the most important aspect as far as this quality by design is concerned. So, these are the points which differentiate between the current approach which we are using and quality by design approach. Next slide, please. Now, what is the background? Quality by design, as I mentioned in the first slide, it is not a new concept. This concept we are updated, and this particular concept has been well explored by the chemical engineering field. Next. So this concept is based on the thought which is given by Joseph Giron, he is a quality expert. Quality cannot be tested in the product, but it has to be built inside. Now, many literature documents are available as far as basics of quality by design is concerned. I'm not going to give more stress on this particular aspect. But why FDA, why regulatory bodies feels important, or why regulatory body feels that we need to implement this particular quality by design in the pharma sector? There are certain reasons that we'll discuss. Next slide, please. Now, if you see the history, now since you are BPharm final year students, you must be knowing about what is NDA, ANDA. So, if you have gone through some regulatory aspects, confirms that might be knowing what is NDA and ANDA. Different manufacturers need to submit this particular applications to the regulators. Now, in around 2000, number of supplements to the original applications have been submitted by the regulatory bodies. And these particular supplements were more than 5,000. Evaluating applications by the pharmaceutical manufacturers, it was a tough task. But in addition to that, in a very short span of time, they have, with manufacturers, they have submitted more than 5,000 supplements. And evaluating all of them, it was a very big task. Then regulators thought that there must be some common strategy. There must be some common platform to address all such issues. And they start analyzing all such type of supplements. Supplement, it is an additional document which has been submitted to the original application by that manufacturer. Now, what supplement involves? They suggest that yes, we got the results during our applications, but we want to make certain changes in the original application. Now, what are the reasons behind that particular change? The reason may be, it may be related with that particular chemist. And they observe that all the supplements that have been submitted by the manufacturers, they were much related with the chemistry part. And none of the supplement was related with the process part. So, the focus of all the manufacturers, they were related with the chemistry. That's why it has been decided that in order to bring all the manufacturers on the common platform, we need to adopt certain science which is already existing, which has been explored by the engineering science, and that may give a proper solution. And there were many debates on it, how to implement it, how we can go for it, and implementation of CGMP, implementation of PAD, and many other aspects are also involved in it. In 2006, along with the introduction of ICHQ2, some aspects have been introduced. In 2008, ICHQ8, Q9, and Q10 have been implemented. Now, this ICHQ8, Q9, and Q10, they form the basis of quality by design in the pharmaceuticals. And this ICHQ8, 9, and 10, it gives answer to the questions which the we can say manufacturer as well as the regulator was facing. And in that journey, it has been observed that in the ICHQ8, what is there, and ICHQ9, what it deals with, we will see in the later slides. And after that, some of the issues that has not been addressed in the ICHQ8, Q9, and Q10, that has been addressed in ICHQ11, Q12, Q13, and now in the Q14. Next slide, please. So, the last point, USFDA is accelerating QD drive, means they are slowly implementing this quality by design concept in the pharma sector, because we cannot change the system suddenly. If you want to implement something new, then you have to implement it in stepwise manner. Since regulators at a glance, if they change the entire regulation system, then it is practically impossible. That's why slowly they are implementing it from 2014 onwards in US market. They have implemented for this generic market. Later on, as far as analytical method developments are concerned, they have implemented some regulations in this ICH 14. Next one. So I just now mentioned about what is the correlation between ICH and quality by design. If you see the basic definition in 2008 and 10, they have given that ICH quality by design, it is nothing but ICHQ 8, 9, and 10. Primary definition: pharmaceutical development, quality risk management, and pharmaceutical quality system. Now, with that initial definition, regulators were not happy. Number of issues have been raised by the manufacturers. This particular definition, which has been given at the start, it was only focusing on the pharmaceutical product development. It was not focusing on the drug substance. What is the difference between drug substance and drug product? Drug substance is nothing but API. Now, what about API synthesis? The issue related to the API synthesis needs to be addressed. And once the suggestion has been given to the regulators, they have implemented ICHQ 11. Now, again, what happens if we are talking about the control on specific part of that particular formulation? What happened to that particular life cycle of that product? So as soon as some issues have been raised by the different manufacturers, regulatory bodies, they are implementing the different sections of drug development through this ICH, and they are now in a process to implement this ICH Q14. Now, what this ICH Q14? This is the objective of this particular lecture. Now, by the time you actually start working on the different analytical method development procedures, or if you start working in the industry for development of certain procedures for analysis of the drugs or some sort of products, then these guidelines will be effective, because the primary date that they have planned was May 23, but still it has been not implemented. But this document is available on the website of ICH as a document for public consultation. Means we can give some, we can say, suggestions related to these particular guidelines. But 80 to 90% of these guidelines will be the same as such, and we can get some new additions to this particular guidelines. Now, we should know what exactly these guidelines are. What is the difference between classical method development and analytical method development? Now, the students who work on this analytical method development aspects, these particular guidelines will be helpful. Now, how they can think in the different direction? Because as sir just now mentioned, working on the different projects, getting new drugs, it is practically difficult. So working on the existing drugs with some new science, that is a new idea as far as pharmaceuticals. So as sir just now mentioned about, if you explore the existing drugs as far as co-crystals are concerned, that will give you a new idea. But as far as analytical method development is concerned, when we were working on, we have published many articles on colorimetric estimation. On colorimetric estimation, I have published 10 articles. But can this particular concept works now? It will not work. I published 4 articles on UV method development and validation. Can that particular concept works now? No, this is outdated concept. I worked on stability indicating assay methods for a particular drug. I published more than 10 papers on it. But can that concept work now? No. We need to work on every time, as sir just mentioned, getting new drug, working with the old concept with the new drug. Practically, it is now difficult. That's why working with the old drugs with some new concept, that is much important. And that's why we need to think about what this ICH-Q14 is and how we can develop analytical method in this particular paradigm as far as quality by design. So ICH-Q14, it speaks about analytical procedure development, and it has been renamed as ICH-Q14/ICH-Q2R2. Previously it was Q2R1. Now it has been renamed as Q2R2, where the revision of existing R1 is Q2R1, where analytical method development and validation has been given. Now, if you see, in order to understand this ICH-Q14, you should know what exactly is Q2R1. So it gives an information about how we can develop the analytical method, how it can be validated, what are the validation parameters, then how we can go for SIOM, that is stability indicating assay methods. Unless and until you have some background about information about this particular aspects of Q2R1, we will not get idea about what is Q2R2. Next one. Next one. Next one. Yes. Now see, in the current scenario, as I mentioned, initial definition was QBD is equal to ICH 8 plus 9 plus 10. Then some of the suggestions have been given to the ICH that yes, this is covering almost the drug development and it is much related to the drug products. But what about the drug substance, that is API? Then they started to implement the guidelines related to the drug substance, that is API. They are focused on the manufacturing of API and they have given some guidelines, and that has been reflected in ICH-Q11. As far as ICH-Q12 is concerned, it is all about the product life cycle management. So, giving information to the regulators about initial stages of the drug development is not sufficient. The drug must, or the product must clear the properties which we are giving to the regulator till the life cycle of the product. And that is the basis of this ICH-Q12. Now, as far as ICH-Q13 is concerned, continuous manufacturing. There is a need to improve the process. If after analyzing the process, if I find certain lacunas in that particular process, there must be scope for the improvement. Continual improvement is the basis for this ICH 13. And as far as Q14 is concerned, there were many suggestions which have been given. Even as a researcher, as a informed student, you also can give the suggestions to the ICH. Because these draft guidelines are available on the website. If you feel that this particular part needs to be removed, this particular part needs to be added, as far as ICH is concerned. ICH draft guidelines are open for all. Any person from any science, he can give the suggestions as far as addition or deletion of some part is concerned. If you suggest deletion of some part, then you must do some scientific proof. If you want to add, suggest to add something, then also you have to do some scientific. And if they are not agreeing with the part which you have suggested, they will give the written answer. Because I have received many suggestions from the ICH as far as some suggestions which I have claimed in a written form. Now, what are the basis of development for this ICH Q14? ICH guidelines, they have been framed not only by a single person, they involve regulators from the different part of the world. They involve different manufacturers who are actually involved in manufacturing. And they involve many of the people who are expert in that particular area. And selection of experts in that particular area, it is the right of the regulatory bodies. Now, based on this quality by design concept has been slowly, you can say, in a phase of implementation to all the pharma sectors. Next slide, please. So, this is a principle of quality by design. What exactly this process? As I mentioned just now, it is a science and risk-based process. It is totally depending on understanding the process. It is totally depending on predefined objectives. Means what I want from that particular process, I need to implement the start. I need to work accordingly in order to get that particular objective achieved. So, understanding the process is the most important component if I want to get over success as far as the process is concerned. If I want to become successful for that particular objective related to the process or a product, then understanding the process, understanding the properties of the product which on which I want to work, that is much essential. With that aspect, the first aspect is defining the objective. Then determination of critical quality attributes. What it is? It is that attributes may be related to the product if I am working on the product. The attributes may be related to the process if I am working on the process. If I am working on analytical method development, what are critical quality attributes? If I am working on HPLC method development, what are the parameters that affect the performance of the method that I have developed? If I am working on some product, if I am working on the tablet, then what are the process parameters that are tricky that needs to be controlled? They have been named as a critical quality attributes. Based on this particular primary concept of quality by design, the same concepts have been applied as far as analytical method development. Next slide, please. Next slide. Next. Because this entire presentation requires 2 hours for me. So as the time has been very past, so only 50 minutes I have received. So then any ICH guideline, this implementation requires suggestions from the end user. End user means he will be the chemist, he will be the formulation chemist, or he will be the pharmacist who is working on the shop floor. Now, some past literature is also required. Past literature means the experts in that field, what type of suggestions they are given in the literature. And when they think that we need to give some guidelines as far as analytical method development is concerned, then they have gone through the different literature from the experts in that field and they have published some data in the literature. So, the next part. So, Oak et al. In 2011, they have published this particular data in Journal of Pharmaceutical Sciences. And he has suggested some guidelines related to the application of quality by design in analytical method. Now, as I mentioned, the basic principle of quality by design, understanding the process is very important. Quality target product profile, DPP is very important. There must be something synonymous to that DPP in analytical method. What is that synonymous term? They have coined a term that is known as ATP, that is analytical target. Then, as far as design space, which is a more, can say, basic concept as far as quality by design is concerned. Design space, it is a multidimensional combination of, we can say, permutation and combination of all the method parameters that are required in order to become that particular process of method analysis. So there must be something which is synonymous to this design space in case of analytical method. And different researchers, they have given different names. And if you see, as per the concept which has been given by Oak et al. 2011, long back, means when the concept of quality by design has been given, at that time, people were thinking that how to use this quality by design concept for the analytical method. We also have worked on that, that part, and we have published many papers because no guidelines were there. If we have the guidelines, we can work according to guidelines. If in absence of guidelines, how we can work? So we must have some references, and these are the references. Using these two references, we have worked on the analytical method development, and still people are working on the same line. Now, henceforth, Q14, it is a standard guideline. You can use that particular guideline. Next slide, please. This is another reference, that is AQBD workflow, and they have given a name, that is Method Operable Design Region, that is MODR, which is synonymous to design space. Again, what is this MODR? How it is important as far as analytical method development is concerned? There are many issues, and even I can talk on this particular AQBD workflow for 1 hour. But still, I am just giving the difference between the application, how QBD concept can be applied to the analytical method development, and how these particular guidelines have been evolved. So this particular concept of method operable design region, again, this concept has been given by George Day. He has published one paper in the same journal in 2013, and he has given the concept of MODR. The same concept has been picked up by ICH. If you see this ICH guidelines, which has been framed in 2019, it's almost 6 years later. ICH, as such, these are not new guidelines, but these guidelines have been compiled by taking the views of end users, by taking the views of manufacturers, by taking the views of experts who have been already worked on that particular area. So these two references have been considered as the basic references as far as analytical method development and we can say compilation of ICH Q14. Next. Yes, this is the content of ICH Q14. Now there are many things that can be explained here. But with the constraint of time, I will specify the things which are important as far as the method development. So this ICH Q14, it is nothing but an amendment of ICH QR1. The R1 guideline Q2R1, which we are using since 2005 as a basic tool for analytical, basic guideline for the analytical method development. This particular guideline has been totally revised. It has been renamed as an Q2R2. And this particular guideline, it is based on the fact that rather than developing the method and validating it, you can go for that particular approach. That the approach that has been named as a minimal approach. But you can go for risk analysis. You can go for risk identification. You can go for risk minimization. The help of control strategy. So overall, the basic principles of quality by design have been implemented in this particular ICH report. Now, what is minimal approach? Minimal approach, they have suggested two approaches in ICH Q14. These are the draft guidelines. These are not final guidelines, but 90% of the same guidelines will be there as a final guideline because very few guidelines, very few points may be reduced from. What is the basic approach? Basic approach is identifying the analytical tool, working on that particular tool, developing the analytical methodology, and validation of that particular analytical methodology that we are using traditionally. And another tool or another approach they have suggested that is enhanced approach. That is a core part of this ICH program. What is enhanced approach? Next one. See, as just now mentioned, there unless and until there are certain limitations to the existing science, new science will not evolve. New science, new concepts are the results of limitations of the existing science. So we were not able to correlate, or we don't have the strong guidelines as far as analytical method development in QD framework is concerned. That's why we were working blindly. As per some references which have been reported in the literature, we were working on the aspects of analytical method development. But if to overcome the limitations of the existing science or existing concepts, if you want to do something new to the pharmaceutical industry, then it must have some sound base. So, with that view, considering the core concept of quality by design, these guidelines have been given, and these guidelines definitely will helpful to address the issues which we are facing as far as analytical quality by design is concerned. Next one. Next one. So these are some of the objectives. Basically, these particular guidelines will definitely address the issues which we are facing nowadays. How to work on it? At the end of this particular presentation, I will use a short view about it, how we can implement these guidelines in our actual work. Next one. That is more important as far as working on the project. So these are some expected benefits: harmonization of scientific approaches, then predefined performance characteristics, that is the main feature. Then enabling the preventive measures means once you identify some of the areas where there is a need to gain the control, then gaining more control on this particular critical quality attributes as far as analytical method development is concerned, that will solve the issue. We can get the performance, design performance and state at the start. That is predefined objective which we are following next. Next one. Next one. Yes. Yes. So the two approaches that have been suggested by the regulatory bodies as far as this ICHQ 14 is minimal approach and enhanced approach. Now, what is minimal approach? This minimal approach is in line with ICHQ outcome, which we are traditionally using. Means what we need to do, we need to identify the attributes that need to be tested. Which particular parameter of analyte under consideration? Analyte means the substance whose analysis is to be done. Which particular property of that analyte we want to measure? That is the most important aspect. Then selecting the appropriate technology and related instrument means which instrument will give the answer of that particular property which I want to test. The third aspect, conducting the appropriate development studies means I have to identify the properties of analyte that I want to analyze. I have to identify the technique which I want to use. I have to identify the required instrument. Then I have to work on the development of analysis. Once I develop that particular method, then next defining the analytical procedure means I need to document that particular procedure and I have to submit it to the regulatory body. So this is the minimal approach. Again, the validation remains a mandatory in this approach because validation is the most important tool as far as supporting the analytical process. Now, the enhanced approach that has been suggested in this ICHQ 14 is evaluation of the sample properties and defining the analytical target profile. As I mentioned, quality by design, it is based on the predefined outcomes. Means what I want from that particular analytical procedure or process that I need to define at the start. And once I define my objective or my expectations from that particular process, I need to work on that particular process in order to achieve the objective from. So analytical target profile, it is a synonymous term which has been mentioned with the UTPP with the original quality by design. Now, this analytical target profile I need to mention at the start, and I have to work on that analytical method development in order to achieve this ATP. Now, as far as method development is concerned, I can give few examples of ATP. I want to develop analytical expensive method for X drug with USP telling, specific USP telling, with specific number of theoretical plates. If I want to work on two drugs, I can claim ATP as developing an expensive method with a resolution is this centimeter, is this time, particular time. So ATP may be varied based on the objective of the product. ATP may vary based on the properties of the analyte. So ATP is the most important aspect as far as enhanced approach and conducting the risk assessment. Now, as I mentioned, very simple approach as far as quality by design is concerned. If you want to work on some product, if you want to work on some process, if you want to get the success of the as far as the process is concerned, we need to gain the control. We need to analyze where the risk involved as far as the success is concerned, as far as the process is concerned. Once you identify the risk, the next step comes risk minimization. For risk minimization, you have to take the help of different statistical procedures. And once you reduce the risk, you can ultimately gain the control of the process. Once you gain the control of the process, you can get a success in the implementation of the process. And the similar, we can say, philosophy we can apply here also. So basically, as far as analytical method development is concerned, enhanced approach is the core of analytical quality by design. And this particular enhanced approach, it has been suggested first time as per the ICH guidelines. But the most important point which I want to mention here is minimal approach is a mandatory approach which has been suggested till now. Is if you want to develop a method, analytical method in distributive framework, you have to use minimal approach, and additionally you have to use either some components of PBD or all components. But again, when we are implementing something, we cannot give guarantee about the success of that particular regulations unless and until they are practically working. Because many guidelines have been entered as a guidelines in practice, but after a few years, 2 years, 3 years, these guidelines have been taken back with certain limitations. And again, some amendments have been there. Since it is very, these particular guidelines are at the primary stage, we require some time whether these guidelines are used or not. Next one, please. Next. Thanks. Yes. So this is the particular diagram they have given in ICHQ14. Now this is the entire course what this ICHQ14 is on. I just now mentioned I want to work on analysis of one analyte, and I must know what property of analyte is very important. What property I want to measure. Then I should know which technology I am going to use in order to measure that particular property. Is whether I am going to use UP, whether I am going to use HPLC. What type of, if I have decided to work using UP or HPLC, then I am going to the next step. Then once I define what are my expectations from that process. Means if I want to work on HPLC, in order to measure a particular specific property of that particular drug substance or analyte under consideration, then what should be my ADP, that is analytical target profile. Then once I define my target profile, then I have to decide what are the areas where I need to do more focus. For that, I have to go for the risk assessment, risk analysis, risk minimization, and I have to plan according to the control strategy. And this control strategy will help us to gain the control of the method. So this particular diagram, it will give a clear picture about how analytical procedure life cycle will work. Now, as far as life cycle management of the product or process is concerned, the process should be effective till the end of the product, means life cycle of that product. So some of the guidelines which will give some information. When I am claiming that this particular procedure will be helpful for determination of the X drug. And once the drug has entered the market, that particular procedure, because of degradation of that drug, it is not working. Then that particular procedure is not as per the ICH report. But it suggests that procedure must be worked till the lifetime of that particular process. If I'm working with the process. So that is the most important aspect, and this will give a clear-cut idea about analytical procedure life cycle. Next one. Now ATP. Again, the definition of this ATP it has been given. So this is a you can say most important component of this ICH14. And as far as I want to use enhanced approach, this ATP plays an important role. Then what is ATP? It is a summary of all the parameters, summary of all the process parameters. If I am working on the analysis of a product, then I have to take the consideration of properties of that particular product. If I am working on one process, if I am working using HPLC, because most of the students, as far as analytical method development is concerned, they are working on analytical method development and validation using these ICH guidelines. Now, in that era, we need to understand what is the property of that particular process. We need to understand which methodology I want to use. And we need to understand how I can implement these particular current ICH guidelines as far as analytical method development. So setting analytical target profile. I can set analytical target profile which may incorporate 2 to 3 components, but working on the academic platform, setting 2 to 3 targets at a time is practically impossible. I can choose only one target that can be achieved, that can be in line with the ICH guidelines which have been mentioned. Next. Yes. Knowledge and risk management is the most important aspect as far as analytical method development is concerned. So the concept which has been given initially in ICH Q8, Q9 and Q10, the same concept has been given here. Understanding the product is important in general QBD, whereas understanding the process of analysis is very important. But understanding the process along with the understanding of the product whose analysis is done, that is more important in this case. What is the difference? As far as the general concept of QBD is concerned, that is quality by design is concerned. So if I'm working on the product, I need to understand fully the information about the product. But here, if I'm working on the process, if I'm working on the analytical procedure, then in order to gain the control, in order to gain the results which I am expecting as per my predefined objectives, as per my ADP, if I want that particular results for during the life cycle of that particular product, then these particular aspects, that is knowledge about the product, that is analyzed, and knowledge about the process is very important. The same knowledge and risk management. And for that, if certain parameters are more risky as far as method development is concerned, I can give the practical example. If I want to work on HPLC method development, during HPLC method development, there are many important aspects as I just now mentioned. If I want to work on the X drug, properties of the X drug plays an important role. That will help me choosing proper column. That will help me choosing proper mobile phase. Once I initially set that particular initial mobile phase condition based on the previous history of that particular drug, then I want to develop the method under QBD environment or using the QBD ICH Q14 guidelines. What I need to do? I need to go for the risk. Means I need to identify which are the parameters which may affect the performance of it. The parameter may be column mount temperature. The parameter may be the type of column, whether it is CL or scale. The parameter may be the flow rate of mobile phase. The parameter may be the lambda max of the drug. Parameter may be the composition of mobile phase. Again, into aqueous ratio. So I need to enlist all these particular parameters. Once I enlist all these parameters, I need to identify which are the parameters which make it more critical. So for that, again, I have to take the help of statistics. And these particular parameters, out of 6 parameters which I identify, they can affect the performance of the method. Then I have to identify more critical parameters that may affect the performance. And these particular parameters have to be named as CQAs. Then my focus will be on this. I have to go for the control strategy. And in the control strategy, I have to take the help of statistics and I will go for permutation and combination of all these parameters which are more risky, and I will develop a method which is we can say more robust, and I will explore the robustness parameter which we are using as a traditional analytical tool method development evaluation. So this is the most important aspect as far as knowledge and risk management is concerned. So what is the basic difference between traditional analytical method development and method development under this QBD framework? These are the most important parts. So understanding the product and process, that is most important as far as analytical method development. And once you gain the control on these particular parameters, you can get the success. Whatever the method you have developed, you have explored the robustness parameter, and that's why spatial validation parameter validation, that is a part of Q2R1, is not included in this process. So once I develop the analytical method under this Q14 guidelines, then validation remains a formality because it is a self-validated. So this is the most important aspect. This is the most important difference between the current approach and the approach which we are using. Next one, quality risk management that I spoke about. Because one of the tools used for quality risk management, again, it will be a big topic of discussion. And how we can go for the control strategy, that is also a big topic of discussion. Then, evaluation of robustness, that is the most important aspect. As I just now mentioned, robustness is a parameter. What is robustness? Some deliberate changes in the method parameters should not affect the performance of the method. This is a good definition of robustness. And this particular robustness has been more focused in this analytical quality by design to the help of ICH2. So we are purposely variating or deviating from some established conditions of that particular method. Some purposeful variations that we made during the development which should not affect the performance. To what extent we can vary that particular method, variation of parameters that has been given as a standard. So we cannot extend beyond the range that has been given. And if the method which we develop, if it is giving the same results as per our expectations, it is not getting affected by some deliberate variations. The method is said to be robust. And as far as minimal approach is concerned, which is mandatory as per this ICH guidelines, we need to explore robustness. If you want to go for enhanced approach, that is approach using quality by design, some of the element is robustness. So using robustness, we can explore that particular data. Next one. Yes, this method operable design range, that particular MODR, which was a concept which it has been given in 2013, that has been taken as such in ICH2, which is synonymous to the design space which has been given in the original concept. So the concept of design space has been renamed in this particular ICH2 Q14. The concept which has been given in ICH-Q8, which is for general quality by design, it has been renamed as an MODR in this particular ICH-Q4. Next one. Next one. Again, planning the control strategy, and as I just now mentioned, identifying the critical parameters and gaining the control of the process. This all depends on how you gain the control of these parameters. Means out of 6 parameters I just now mentioned, only 3 parameters are important: flow rate, column on temperature, and dampness. If I will control on these 3, then if I will go for statistical evaluation of these parameters and effect of the response may be varied. Response, if I am working on the single drug, response may be USP dense. If I am working on a single drug, retention time may be response. If I am working on the 2 drugs, resolution between the 2 components A and B, it may be the response. So response may be varied based on the type of analysis that I am using. And while planning the control strategy was not a part of quality other than the development. It's the part of this particular enhanced approach as far as quality by design is concerned. And that's why Q14, it will be you can say implemented shortly. Expected was in May 2023, but we are expecting month to month the final guidelines to be available. And these particular guidelines will be definitely can be applied by the people who are working on analytical method development in academic sessions. Because as I have just mentioned about choosing a topic in the different areas of research. Because MPharm research, according to me, is just an introduction to research. If you want to know the definition of research, just reading the definition and writing the definition on the paper, it will not work. So there are many definitions which have been given by researchers. Research, it is nothing but find something new. Research is nothing but addition of knowledge to the existing stock of knowledge. Research is nothing but giving explanation for the already existing process. That process is already existing, but we are giving the scientific explanation first time. So as far as MPharm research is concerned, working completely on the novel topic is practically difficult now because of limitations of the infrastructure. Infrastructure limitation, it is not the issue related with the private institutions, but it is related with the issue of all the institutions. If you think about the researcher working in the NCL, he may have some analytical tools which even cannot think of, but he may not have another analytical tool which he requires. If I want to go for single picture analysis, the NCL researcher can think about it, but I cannot think about it. So just to mention about the importance of single picture analysis. But we cannot blame the system, we cannot blame that we don't have the infrastructure, we don't have we have the limitations. But identify the research problems as far as the research problems that will address the issues as far as new concepts are concerned, concerning with the new old drugs. This is something I mentioned here. Working on the old drugs. If you have one drug and already one method has been developed using the traditional approach. If I can work the same method or same drug, sorry, not same method, same drug under PUD framework, that will be addition for them. And coming era will be the era of quality by design. Now nobody can escape from the concept. Those who want to work in the pharma industry. Initially, they were thinking that since the concept is new, industry will deny it in 5 years. I heard this particular comments from some experts from the industry in 2014. But now they are attending the different workshops based on the quality. The reason is that particular concept has not been implemented by one manufacturer. It has been decided by the regulators. Regulators are slowly increasing their eras of work. They have initially implemented for one section, the section. They have started to implement for APIIR section. Then they have covered now analytical method development. So all sectors of the drug development, they will cover shortly. And on one fine day, quality by design for drug development will be managed. So as a young pharm student, as a B Pharm student, you should know what exactly the concept is. The very basic objective of this particular presentation is to make aware about yes, ICH-248. This is coming and what it contains, how it differs from the existing ICH guidelines, what it involves. Now, for young pharm students, how we can work using these particular guidelines on the existing drugs or on the existing combinations which are already there in the market. Choosing combination, as the number of young pharms are increasing, number of people are working. There was one time we have published many articles on a single drug with 2 different methods. If I develop one method, I will publish one. If I develop second method, I will publish because drugs were limited. Number of people who are, and basically what happens, working on the research topics and working on the novel topics is difficult because there is a tremendous competition among the researchers for new ideas. The idea which is new today becomes old tomorrow. If I just now mentioned, I published many articles on colorimetry in 2002. Simple colorimetry that we perform as a part of glucose estimation in cooling pools. We have published many papers, but we cannot work on such topics nowadays because we have to move along with the development of science and we need to share our findings with the science. That is very important. Working along with the development of science, giving or adding something new to the area of science is very important as far as research. That's why the very basic objective of this particular presentation is to make aware about what this ICH-248 is, how it differs from ICH-2R1, and how we can implement. Because if I took one case study, it would require 1 hour. So if you face any problem in we can say implementation of analytical quality by design or development of analytical methodology in quality by design, you can approach me at any time. So go to the conclusion slide. Next one. Skip 2 slides. Yes. There are many things that I want to tell you because of restrictions of time. I could not focus. Yes. Next. Conclusion. Next one. Yes. Yes. Now, with this ICH 214 regulators have done their job. People were asking how to work on analytical method development. There were many letters that have been sent by many, and these letters were also open. And many researchers suggesting that you mentioned about the basics of quality by design in ICH today. You mentioned about APIIR in ICH 11. You mentioned about life cycle management. You mentioned about continual improvement. But what about analytical quality by design? And with that view, in 2018, they have given one concept paper. Before implementation of any ICH guideline, 2-page document is always published on the website of ICH. This guideline will speak about what will be the face of this future phase. And with the help of this particular concept paper, we need to submit some suggestions. And suggestions may be from end user, he may be the analyst who is working in one lab, he may be any person who is related to that particular field or not related to the particular. Surprisingly, it has been noticed by ICH lab, they are mentioned on their website also. Some of the suggestions have been given by the person who is working on analysis of different agriculture. But he's working on agricultural development, he's working on chemical and analytical development. And they were using certain strategy. Why we cannot use same strategy as far as pharmaceutical and analytical methods. Then such guidelines, such suggestions have been taken into consideration and they have implemented. Now, though these particular guidelines have been implemented, not implemented, but they have been published as a draft guidelines, and in coming time they will implement it. But I mentioned in the last point, though the initial approach or the we can say minimal approach is mandatory, and we can take some of the elements of we can say enhanced approach. That is also a mandation that has been made by the regulatory body. But I have given a question mark on the roadmap because whenever any guideline will be published, when they actually implemented in the laboratories, there are many issues that will be faced by the actual end user, which the analyst will work on it. And the feedback from the particular end user is an important goal. When I'm saying that these guidelines have been implemented and these guidelines are successful, I cannot make this particular statement because implementation or complete implementation will be there only and only when these particular guidelines are successfully used for more time. The definition of more time is the lifetime of the product. So, though the guidelines have been implemented, the coming time will decide whether it is a real roadmap or not. So, this is all about what I want to convey. There are many things which we can, I can highlight, but because of the restriction of time, I have we can make that particular discussion very short. So, anything if you want to ask, you can ask. So, again, as I mentioned, this particular review has been written by a young farm student like you in 2017. It has been published and it got very good citations, more than 250 citations. What are citations? Citations show the utility of that particular argument. This is the data that has been presented in the review article that is useful, that has been used by many researchers. And surprisingly, I can say that few of the articles that have been referred, almost more than 20 articles, which has been used as a reference article for their writing. It involves top 10 articles of the pharmacy, International Journal of Pharmaceutics, European Journal of Pharmaceutical Sciences, DDIP. They have used this particular article because we have given some guidelines. This particular article has been written in 2014, where very few information on analysis of application of we can say quality by design analytical methodology. And that particular guideline we have made, we have worked, and in 2014 only we have published one paper. Next one. Yes. We have published it in the use of systematic approach for the development of APIIR method for estimation of root cause. No guidelines were there, but considering the two important papers that I showed, which were these articles were the base for this ICF guidelines. We use same principles that were published in this analytical chemistry. So this particular book we have written in 2014. Means hardly few articles about the basics of quality by design. If you think about, means only ICH. If you see the definition of QBD, I have given the definition that is 8 plus 9 plus 10. There was no 11 at that time. There was no ICH 12 at that time. No 13 at that time. So now again in the second edition we are improvising it, and the new definition of QBD 8 plus 9 plus 10 plus 11 plus 13, and after implementation of Q14 it will be, so there will be a complete definition which we think. Now there may be some issues for implementation that will be addressed in the future. Next one. So again, once again, I'm thankful to the coordinators of the conference. Again, thankful to our principal who allowed us to interact and coordinators gave an opportunity to interact. Thank you. Anything you want to ask, you can ask. They usually the lecture before lunch and after lunch. This is the always whenever I presented, in these 2 slots I got the opportunity. So I always enjoy it because see, if you have been prepared for 2 hours, then if you get less time. So, but if you have been prepared for 1 hour and if you have not to do it for 2 hours, that is quite difficult. Okay, so still I have shared my presentation also. If you want, you can take that question. Thank you.
Thank you so much, Dr. Jayaprakash and Shetty sir. Now I request Dr. Dhanashri Tumar ma'am to present certificate of appreciation to respected Dr. Jayaprakash and Shetty sir.
I request everyone to stay 5 more minutes.
Maybe.
After this, we'll have a short break. Then we'll gather back here at 2:15 p.m. for another round of sessions. Thank you, everyone. We'll gather back here at 2:15 sharp.
Once again, a very good afternoon to all the delegates present here. I, Sakshi welcome you again on the behalf of our Raisoni University, Saikheda. As we all know, today is the second day of our international conference, APIIR, that is Approach to Pharmaceutical Insights through Interdisciplinary Research. So, without delaying, let's welcome our today's guest of honor, Dr. Nitesh Kumar Jain, sir. Respected Nitesh Kumar Jain, sir, is a professor and head, Department of Pharmacology, Modern Institute of Pharmaceutical Sciences. Sir has more than 15 years of experience in academics and research. Sir has 9 Indian patents, 2 design patents, and copyrights have been issued. He has been awarded with many prestigious awards like Eminent Professor Award, Visionary Principal Award, Excellent Teacher Award, and so on. We feel extremely grateful to have you here, sir. Now, please welcome Dr. Nitesh Kumar Jain, sir, with a huge round of applause.
Hello. Very good afternoon to one and all. So last evening of the session, I had to play it. Actually, I have a lot of responsibility to do. आपको जगाना भी है मुझे. So how was your lunch? Not look sound. How is the joke? How is the joke? It's very difficult. Now I have to say something, right? So, good afternoon, one and all. First of all, I am very thankful to Honorable Management, Raisoni University, Dynamic Dean, Dr. DC Sahu sir, Registrar sir, for giving these opportunities as a speaker in this conference. So from yesterday, you have listened many talk from the chief guest of honor. Today, we have the wonderful scientific session by our two eminent speaker, Lahoti sir from Aurangabad. So in this last scientific session, because being a pharmacologist, being a toxicologist, so now things come to me to check the biological activity of all the session. So here I am, basically I will conclude all the session, and I am taking here artificial intelligence. This is a very interesting topic nowadays. After completion of B.Pharm degree, you will go into the market. So definitely you must be competitive with the technology, right? So in this particular session, I will moreover focus on AI in pharmacovigilance and toxicovigilance. So before going to start the session, as this is the last and you have taken a lot of lunch, lot of food, right? तो एक डीसी साहू सर कभी डांटते हैं आप लोगों को? कभी नहीं डांटते? डांटते हैं? ठीक है, तो उस डांट के लिए, वो डांट कहीं ना कहीं आपके फ्यूचर के लिए होगी, और उस डांट पर एक रूठने मनाने वाला जो एक बहुत खूबसूरत सा सॉन्ग है, जो कि मुझे जगाना भी है आप लोगों को, तो मैं कुछ लाइन है उसकी आप लोगों के लिए गुनगुनाना चाहूंगा। तो दोस्तों, लास्ट सेशन है, वी नीड योर सपोर्ट, क्योंकि मैं बड़ी दूर से आया हूं कहीं ना कहीं आपसे इंटरेक्शन करने के लिए। तो जब कोई टीचर डांटता है तो कहीं ना कहीं उसमें आपका फ्यूचर, जो एक ब्राइट फ्यूचर होता है, उसकी शुरुआत लिखी होती है। तो रूठ के हमसे कहीं जब चले जाओगे तुम, रूठ के हमसे कहीं जब चले जाओगे तुम, हमने सोचा था नहीं कितने याद आओगे तुम, रूठ के हमसे कहीं जब चले जाओगे तुम। मैं तो न चला था दो कदम भी तुम बिन, मैं तो न चला था दो कदम भी तुम बिन, मगर मेरा बचपन यही सोचे हर दिन, लौट के जाने वालों अब कहाँ जाओगे तुम, लौट के जाने वालों अब कहाँ जाओगे तुम, हमने सोचा था नहीं इतने याद आओगे तुम, लौट के हमसे कहीं। थैंक यू सो मच। ये सॉन्ग इसलिए था कि कहीं ना कहीं डांट, जब हम कोई इंटरव्यू फेस करते हैं और जब कहीं हम एक अचीवमेंट्स को पाते हैं, तो हमें ये चीज याद आती है कि आज काश हमारे टीचर ने या डांटा ना होता तो हम यहाँ ना होते। Now we come to the scientific talk. So my dear students, I am talking about the artificial intelligence. Artificial intelligence, nothing, all of you are using, by using your Android mobiles applications, all of you are using WhatsApp, Facebook is and all, right? But being the scientific community, we are the part of drug discovery, right? So drug discovery is a very complicated process. We know all about it, and we are becoming pharmacy of the world, right? We are the superpower of pharmacy. We have seen COVID-19 pandemic. We all are the witness of this particular medical emergency. And we have seen the hard work of our pharmacy officer. Previously we know as pharmacist, now what we become as pharmacy officer. So one thing you must proud yourself. Doctors give life to patient, but with the help of medicine. But we all pharmacy officer give life to medicine. So actually we are the father of medicine. We must be proud to be a pharmacist. We are the specialists of medicine, right? Agree? So first of all, please clap for all of us. Because we are the part of novel profession, we are the part of healthcare system. So for that, here I am giving presentation on artificial intelligence. So all of you know about the pharmacovigilance and toxicovigilance. Here we are talking about the safety of medicine. Do you know about the pharmacovigilance? What is pharmacovigilance? Anyone can explain? Yes. We are talking about the ADR, that is adverse drug reaction. So in this particular presentation, I would like to highlight there are many things has been taught by our eminent speaker from both the days. They have talked about the formulation, quality assurance, synthetic chemistry, right? But now we are coming to the conclusive part, right? Because we have to finish now and the last pillar of this evening. So I want to touch your knowledge about the toxicity of compound, right? So if we are looking for sustainable development of the product or maybe organization, so always we have to think about the toxicity of compound, right? Next, please. This is the one quote which I really love. Find something you love to do. You will never have a work in a day. आप यदि जो काम करते हैं उससे प्यार से कोई काम करते हैं तो कभी भी आपको थकावट महसूस नहीं होगी. तो we are here in a very noble profession. जैसा कि हमारे चीफ गेस्ट जो APTI के प्रेसिडेंट ओवेकर सर ने भी कल बोला कि we need your dedication, commitment towards the profession, right? We are here as a speaker just to motivate you and what can be the research findings as Honorable Lahoti ji sir suggested many things about the co-crystallization, quality assurance by design. So there are many things which is coming nowadays in the form of artificial intelligence. Okay? So people have misconception that artificial intelligence can replace us. Not possible at all, right? Because that artificial intelligence basically a machine learning. So software programming, right? So whatever concept we are giving to the machine, accordingly that machine will give result, right? That cannot be anyhow replace the human manpower. Whatever things we have, whatever emotions we have, so definitely we have to follow it, right? Next, please. So what is AI actually? So artificial intelligence is a stream of science related to internet, machine learning, mainly intelligent computer programs, which proves result in their similar way to human attention process. This process generally comprises obtaining data, developing efficient system for the use of obtaining data, researching defined and appropriate conclusion and self-correction and adjustment. Okay? Recently, AI technology become a very fundamental part of industry for the use of application in many technical and research field. So ultimately, in regular practice, all of you are attending your classes, practicals, but we have to moreover focus on software programming now. So already you have some software, maybe all of you are in B form, but when you will go into the PG, so definitely you have to attend. Many you have to learn many software about like USAR. Sir has discussed many things about the docking, computer-aided drug design. So what happened in drug discovery? There are two phases. First one is preclinical and second one is clinical. So previous days generally we required 14 years, right, for complete drug discovery. Hai or it's very easy to take that particular medicine in a second. So it's required many things which we have to optimize for launching the products in the market. So here I want to mention how many phases are in clinical research? All of you are wrong. 5. Now 5 phases are in clinical research. One phase 0 added recently. So that is called AI. Right? Artificial intelligence is books are not revised every year, every month. So we have to connect it with the AI tools, some recent data, some recent websites about what kind of clinical trials are going on, what kind of things marketed, what kind of new technologies are coming. So how AI came? This is the history of AI. In 1950, the time when it was started. 1955, John McCarthy, quantum artificial intelligence. Then in 1974, computer become faster and affordable. Then 1980, the year of artificial intelligence, and the 2000 landmark of the AI establishment. Next, please. So this is the history about the AI. of how we can implement AI, especially in drug discovery. So artificial intelligence has received increased attention in recent years. AI is a rapidly advancement technology made possible by the internet that may soon have significant impacts on our everyday lives. Every day we are using so many apps are coming. We are ordering food from the Amazon or other things, other websites. We are using it, right? So most of the newcomers are using already AI, but we are here to make a future. So what kind of AI application we may use in our profession to make our bright future? That I will discuss here. Next, please. So AI focusing in producing intelligence modeling, which helps in imaging knowledge, cracking problems, and decision making. So recently, AI play important role in various fields of pharmacy like drug discovery, drug delivery, formulation development, polypharmacology, hospital pharmacy, drug discovery, and drug delivery formulation development. So various artificial neuronal techniques we are using here, deep neuronal networking and recurrent neural networking are being employed. So several implementation of AI, especially discovery, have currently have been analyzed and supported with the power of technology in quantitative structure activity relationship and the QSAR. In addition, de novo design promote the invention of significantly newer drug molecules with regards to design and optimal qualities. Next, please. We know the key log theory, right? This is the perfect example. So if you are discovering a new medicinal agent, I think all of you want to become a scientist, definitely. So for any disease, firstly we have to identify the target. So in target, what we have to see the quality of target, nature of target, what kind of shape he has. So if we have a key, if we don't have a lock, can you save your home? Right? So the key is a drug and lock is a receptor. So without receptor, key can't do anything, right? So for the proper biological activity or pharmacological action, we need target, we need receptor. Right? So this is key lock theory concept that is already you know about it. Next, please. This is completely drug discovery process. What are the advantages of AI in drug discovery? Just I want to explain it. Previously, that is around 14 years, but now it's 7 years. But in case of COVID-19 pandemic emergency, what we have seen, the vaccine came within 1.5 years, right? Is it right? How it is possible? With the help of AI only. What AI can do actually in drug discovery, if something running in your mind, right? कि मैं ये कर सकता हूँ, मैं ये कर सकती हूँ. But it is, whether it is possible, basically, physically, or with the help of sources, whatever we have. So AI can help you out by giving data related to that particular segment. So AI can give authentic data to you. If you are searching on Google, that is not the authentic data about the drug discovery, right? There are many plants, medicine. If you will search on internet, so definitely all the plant having anti-diabetic activity, anti-hypertensive activity, there will be no authentic data. So AI definitely will be helpful for all of you, but we have to search authentic data on that. So there are some reliable sources. We will discuss about these AI tools. Next, please. Next. Drug discovery process, as we talk about, if we are looking for compounds, basically what we do, synthesis, or maybe we can obtain some compounds from the plant source. Or we think about that herbals are always safe, right? But this is completely a misconception. There are many herbals, medicine which can cause various kind of chronic toxicity like neurotoxicity, nephrotoxicity, hepatotoxicity, and teratogenicity also. So this is completely a misconception. And nowadays, every PG and PhD research scholar moreover focusing on herbal medicine. But as far as my thinking is concerned, definitely herbals are having in trend. But we have to think about the toxicity of herbals. If we are looking for sustainable development, if we want that product will forever in the society for the welfare of human being as far as health is concerned. So ultimately, if you are making some formulation, if you are analyzing some formulation, apart from the pharmacological activity, we must go for the toxicity study. There are three types of toxicity study generally: acute, subacute, and chronic. Acute is generally basically we conduct for 7 days, subacute for 28 days, and chronic up to 6 months, right? So what is the motto of this toxicity study? I will explain in new phases. But in case of clinical trials, as you are seeing the advancement, how many instruments you have used for HPLC UV? Okay, very few. But this is my personal suggestion. When we have done BPharm, MPharm, the instrument was old, right? But now upgraded versions are coming in the labs, right? Now we are seeing the HPLC fully automated. But in a single time, we can put 100 samples on HPLC and we can get a result immediately. So that is called AI. AI, nothing else than the software integrated system. Whatever algorithm we have put it in that particular instrument, definitely that will save the time for drug discovery as well as cost also. As sir told us in the lecture, millions dollars we required for a drug discovery process. So AI can give fixed target, AI can give actual data. If things has been already done in a proper authentic center, we didn't want to repeat, right? That can save the money, that can save the target. So there are many researchers who are doing research on right now diabetes, on cancer. Ultimately, we have to find out the research gap where we are lacking. So that particular research gap, if we are giving to all of you, you can work on that and we can have some product which can be useful for the society. Now the things must be oriented to the society. If we are doing research just to complete the degree like MPharm or maybe PhD, maybe postdoc, that cannot have a better result, right? If you want to become a great man, especially in the field of healthcare and pharma, so you have to do something very innovative which can have direct relation with the society healthcare system, right? Next, please. This slides I want to highlight. This is the example of AI, right? If I will say something, what is targeting to the cancer, hope you will not understand. बिकॉज कहते हैं ना जो आंखों को अच्छा लगता है वो किसको अच्छा लगता है दिल को और जो दिल को अच्छा लगता है वो दिमाग में हमेशा रहता है राइट तो दिस इज द बेस्ट एग्जांपल इफ यू आर टॉकिंग अबाउट द टारगेटेड ड्रग डिलीवरी सिस्टम व्हाट इज कैंसर abnormal growth of the cells right why there is no complete curable treatment for cancer now we are having a very advanced technology we are talking about the AI, but still we are having issues or we are having a problem that we are not able to cure cancer completely. Why? Definitely with the help of advanced technology, with the help of chemotherapy, radiotherapy, immunotherapy, we have increased the life of the cancer patient up to 10 years. They can survive in the life. So that is a good achievement of the science. But still we are not having complete curable treatment. Why? What are the reasons? Toxicity, adverse effect, side effect of that particular therapy. Because what happens generally, the metabolic pathway of cancerous cell and normal cells are almost same, right? Do you know about the central dogma of life? DNA to RNA and RNA to protein means transcription and translation. This is the central dogma of life. So if we are targeting any anti-cancer agent, generally they interfere with this transcription or maybe translation. तो जो पेशेंट है वो कैंसर से नहीं मर रहा बट जो नॉर्मल सेल्स का destroy हो रहा है with the help of treatment whatever giving to the cancer patient I will show you. So this is the example of targeting. If we can target the cancer cell completely without disturbing the normal cell, then only we can win on the cancer. So you have to think about the targeting, how we can go for the targeting. We can't say medicine, you have to kill only cancer cells. Please don't touch normal cells. Can we? Not it at all, right? Because we have to create a property of that particular drug that can affect only the cancerous cells, not normal cells. Then what will happen? Toxicity will be less, side effect will be less, then patient can survive more and more, and hopefully he can live better, right? So if we are putting the chemotherapy, it can target all the organs of the body. If we are having a targeted that can kill on the cancerous cells. Next, please. Here you can see the concept of targeting. All of you have read about the plasma membrane. Every drug has to cross the plasma membrane if you want proper absorption or maybe therapeutic response, right? So size does matter definitely. If we are having the large size particles or maybe drug, it cannot easily cross the plasma membrane. Here you can see the both cells, normal cells and target cells, right? You are able to see the movement of the drug, but larger molecule are not able to cross the plasma membrane. But in a second point, if we are taking, we are going for the inverse targeting, and what we have made nanoparticles, nano carrier. So definitely they can easily cross the plasma membrane, but here they are killing both the cells again, cancerous cells and normal cells, right? In third, if we are achieve this concept like targeted nanoparticles, then what will happen? They will only kill the cancerous cells. They will not kill normal cells. Then what happened? Toxicity will automatically reduce and we can give better life for the cancer patients, right? Is it clear to all? समझ में आ रहा है? I can understand आपके टर्म होता है फार्माकोलॉजी में टैकिफाइलेक्सिस रिसेप्टर सैचुरेटेड तो नहीं हुआ है because since morning you are taking scientific section तो रिसेप्टर थोड़े से स्टिमुलेट करने की मैं कोशिश कर रहा हूं तो is it clear to all the concept of targeting क्या है you can think about it right next please here you can see the APIIR effect next now you can see this one. This particular cells have a specific receptor for that particular medicine. What will happen? It will bind to the only targeted cell, that means cancerous cells. Another one, normal cells don't have any kind of site, so that particular anti-cancer agents or medicines will not bind to the normal cells. What will happen? What will happen? Toxicity will less. Ultimately, normal cells will be safe. Your immunity power will regain. Your immunity power will not disturb. So this is the ultimately goal of every scientist who are working on anti-cancer drugs. So hope you can have some clear picture. If you want to work on anti-cancer agents, so we have to moreover target on the targeting, right? If we can save the normal cells, definitely we will be able to cure cancer completely. Is it right? Is it right? Okay. Next, please. Next. Now we are coming to the toxicity. This is moreover, we are made up of chemicals and we consistently interact with the chemicals made by the nature and man, right? And all chemicals have the ability to be toxic, right? Next, please. We must breathe, drink, eat chemicals to stay alive. And everyone has the limit. One terminology, one scientist, one idiom which is remembering. Excess of anything is bad. I think किसी भी चीज की क्या होती है? बुरी, right? So if you are taking something in that specified quantity, that will definitely give you the benefit. For example, milk. If you are taking milk, that is good for health. आपके पापा, मम्मी, मौसी, मौसाजी सब कहते हैं, right? But if you are taking excess milk, what it will cause? Constipation. So ultimately, quantity of that particular drug, chemicals, agent will decide whether it will be beneficial for you or whether it will be toxic for you, right? So everything, whatever we breathe in environment, we are taking so many pollutions and all, drink, eat. So all chemicals have ability to be toxic. So the only thing that matters, that is the quantity of that particular compound, drugs or agent, which can have toxic effect. Next, please. If we are talking about the vitamin C, this is a perfect example so that you can understand excess of anything is bad. If we are talking about the vitamin C, you can take from the orange and pomegranate. Very little if you are taking, that is essential for biochemistry of your body. If you are taking 50 mg, that is the daily requirement of your body for physiology, means for function, right? If you are taking 500 mg, that is useful for common cold, right? Now, if you are taking that vitamin C, 500 mg, that will be beneficial for common cold. If you are suffering from common cold, you can take orange, you can take pomegranate, definitely that will be beneficial. But if you are taking 5000 mg, that may cause what? Kidney stones. So this is the fundamental behind the toxicity. Mean ultimately we have to work on therapeutic index. How many are in final year? B form final year and M form? Okay, okay, great. Anyone can explain what about the therapeutic index? Have you seen one graph AUC? There are two lines MSC and MEC. So what we have to do? We have to maintain the dose of that particular drug in between. MSC means maximum safe concentration and minimum effective concentration. Okay? If you are giving less dose, there will be no meaning of that particular dose because it will cause no effect on the body, right? But if you are giving some excess amount, it will go beyond MSC that cause toxic effect. Is it clear? So ultimately, TDM means therapeutic drug monitoring. This is the tool which is basically the need of every clinical research organization. In clinical research organization where we have to conduct the clinical trials, they are basically working on TDM. What can be the therapeutic dose, what can be the toxic dose, and what can be the dose for MEC. So according to, they have to decide what dose will be better for the human being. Next, please. Here you can see the dose determine whether a chemical will be beneficial or toxic. Aspirin 300 to 1000 mg, that is the dose, beneficial dose, right? Therapeutic dose. But if it is go beyond 1000 to 30,000 mg, it may cause severe toxic effect. Same with vitamin A and vitamin C. Now, next, please. Here you can see the dose response curve. If you will come for PG research, so before going to the clinical trials, we have to do a study on animals, that is called preclinical trial, preclinical research, right? So what we have to do? We have to do synthesis of the compound. Either we can take some compound from the plant extract, right? Then we have to go for the lead optimization. You know about 5000 compound generally enter into the preclinical phase. और उसमें से यदि 5 कंपाउंड भी आपके क्लिनिकल फेज मतलब फेज 0 में भी जाते हैं तो दैट इज कॉल्ड सक्सेसफुल रेट ऑफ दैट पर्टिकुलर. So what AI will do, we have lot of data, there are many scientists who are working on it, right? There are many universities, many academic institutions, many research institutions who are working on it, right? So there are some authentic source what they keep the data, they have the ability to analyze the data, right? If you are taking some plant extract, there are some phytoconstituents which are responsible for hepatoprotective activities, some are responsible for antihypertensive activities, some are responsible to treat the kidney stone, right? So with the help of artificial intelligence, if you have a mind like, sir, I have to do some work on diabetes or diabetic complication. So firstly, you have to do extensive literature survey. Generally, that is the boring part of the research. And according to me, this is the most important part of your research. If you are doing proper literature survey, then only you can find a proper research gap. You will have something different. जैसा कि सर ने बात बताया कि things must be innovative in nature. If you are going for the patent, if you are looking for some PhD topic, so we must find out the research gap. What others have done and what things are remaining on which we or I can work, right? So this is the example of toxicity study, whatever I am talking about in animals. I will not go in deep, but noel means there is no toxic effect if you are taking 10 rats. If there is no toxic sign, toxic effect, no, then LT10, that means lethal dose 10 means the dose which can kill the 10% of population. Suppose if we have taken 10 rats, or maybe one rat will die, that is called LD10. But ultimately we have to calculate the LD50 to determine the therapeutic dose. पता है आपको 110 हम लेते हैं जो भी LD50 आता है, और highest dose especially for the herbal formulation, that is 2000 mg per kg. So if we are giving 2000 mg per kg to 10 rats, suppose that is toxic. If it is able to kill more than 50%, like 6 or 7, we will reduce the dose, we will come to the 1500 mg per kg. If 1500 mg per kg is safe, we can take 110. 110 is what? 150 mg per kg for animal study. Then double of this dose to optimize the response, we can take 300 mg per kg or maybe 400. Then we will check the study on animals. If we are getting good result in preclinical research, then only we can go for the clinical trials. So clinical trials is not very easy. Firstly, we have to submit that entire data to FDA. They will review entire protocols. They will cross-check whatever results you have obtained. Accordingly, they will go for clinical trials conduction in all. Next, please. So AI, we are using APIIR, especially in the formulation development, analysis. As I told you, there are many analytical equipments coming day by day with advanced technology, fully automated HPLC. And in our time, we have to do something on this. Definitely, we have to do manually all these things. Now we have software to check all these things in a limited time. That's why drug discovery complete time has reduced from 14 to 7 years now. So this is a good advantage of AI, whichever we are using. As sir told about the QBD, so this is a new concept. So ultimately, after completion of your BPharm degree, or especially if you are going in industry or if you are going for higher education, we must be updated with the latest technology. This is the need of era. We will not get updated. Definitely, you have to face consequences. Because in pharma, almost jobs are in the private sector. So degree can give you the entry, but your skills, your knowledge can give what? Surveillance. Okay? Next, please. So if we are talking about the global AI in drug discovery market projected to reach USD 1434 million by 2024. From USD to 59 million in 2019. So at the growth of the artificial intelligence in drug discovery market is primarily driven by the factors such as a growing number of cross-industry collaboration and partnerships, the increasing need to control drug discovery and development costs and reduce the overall time. So this is the main motto of AI. So don't be confused. We are not replacing by the AI, but we must be updated. If you want to get higher position in the industry, definitely industry, what industry want? What industry want? They want more income, right? So if we are working on AI, if we can give some sustainable growth development, if we can give better product to the industry, the growth of the industry, definitely you will also grow with the industry growth. Next, please. So drug design application in AI, you can see drug discovery process. First, we can talk about the target identification, prediction of PPIs, prediction of protein folding, what we need for the drug receptor interaction, right? So any drug, whatever we are making, whatever we are giving, we need receptor. So study of receptor is must. We have to know about the identification of targets. So AI can basically help to identify the target. Target means protein, maybe nucleic acid, maybe molecules. So hit discovery, drug reproducing, virtual screening, activity scoring, QSAR. Do you know about the QSAR? Quantitative structure activity relationship. Means there are many functional groups which are responsible for a particular activity. So if you are focusing on the research and if you know about which functional group is responsible for which kind of biological activity, so this is a very simple thing. You can search on the reliable resources by the AI tools, and on that basis you can decide your molecule or maybe compound for the research. Next, please. Next. Next. How AI can use in drug discovery? So traditionally, if we talk, traditional drug discovery method are target driven. Matlab, if we know about the target, then only we can start the research. But especially in AI, we can search the target, possible target, we can make a hypothesis and we can start the research, right? So this is the most advantage things you screen for a small molecule that either interact with its effectors, function in the cells. These approaches work well for easily vegetable target that have a well-defined structure. Still, we don't have treatment for AIDS. Why? Because AIDS virus is just like a bacteria, right? बार-बार अपना getup बदलता है, इसलिए we are still not able to cure completely AIDS. So, however, these methods are extremely limited due to the complex nature of cellular interaction as well as limited knowledge of intricate cellular pathway. Next, please. Next. So artificial intelligence can overcome these challenges by identifying novel interactions first, then an interfering functional importance of different components of a cellular pathway. Right? So AI utilizes complex algorithms. As I told you, whatever we think, whatever we thought about the drug discovery, whatever inputs we are giving to the software, to the equipment, that result will come to you. Right? Like in case of industry visit, how many of you have done industry visit? Now in industry, if you will visit, you will see lot of automation, right? If you are putting the powder form and ultimately you are getting end of the tablet, capsules, whatever it may be, packaging and all. So this is the advancement of AI, or especially IPQC, in process quality control. They have put in many machinery, softwares and all, so regularly they can check the quality of the product. If your product is going to destroy in any stage, we can stop production there only. So what will that will save the money as well as the time for the production. Next, please. So many companies are currently collecting data and increasing their database in order to reduce the time taken for the development and to reduce the cost to do so. So this is the most important thing which I want to highlight. Ultimately, searching the profile on MB, right? For what? Better, you have to study about the AI tools. What's running in the market? What are the main things which we can focus? If we are going for any company for interview or for any kind of training, we must know about the company products, company planning, so that they can choose you, right? Ultimately, you must clear about your basics. Then company have their own SOPs, they will train you, they will develop you. but you must know about the basics, right? So the more clinical and genomic data oncologists have, the smarter decision they can make about the drug usage in any given circumstances. So these such companies understand that AI can save the cost in developing drugs, and AI can incorporate it into the various drug development stages, such as designing new drug after incorporating and analyzing data set for identified lead molecule. Okay? Means if you are thinking about to do some research on a specific field, भारत विविधताओं का देश है, right? ना सबको समझा जा सकता है, ना सबको समझाया जा सकता है, right? So everyone has their own choice. किसी को डायबिटीज पे काम करना अच्छा लगेगा, किसी को हाइपरटेंशन पे, किसी को कैंसर पे. So for that we have to search on different engine. What can be the possible new target for achieving a new milestone in the drug discovery? So my dear student, AI can help you in many ways, and definitely this is a good thing. In our time, we have to do more practicals about the toxicity, about the solubility, and all, right? But now with the help of AI, you can save your time, you can save cost of the drug discovery also. Next, please. So AI can predict its reaction with the biological target also. In silico, in computer simulation, this helps scientists to eliminate poor candidates and increase the chances of obtaining and using good candidates for the research, reducing the chances of drug failing during the clinical trials. So definitely most of the AI tools we can use in the preclinical phases so that we can go for the lead optimization, right? There are many students. If we want to choose one or two, three good candidates, so we have to set some parameters, right? That will be easy to work on it. Next, please. So application of AI, there you can see some AI tools. I will tell the name. So they are basically used for the targeting, to identify the targeted molecule. So phenomic spectra and atomized, TUBAR and MED. So these are the some AI tools in which they have the important and authentic data. Right? Next, please. Benefits of AI, please skip. Now we come to the last part, limitations and the disadvantage. Next. These are the drawback of applying AI in the drug discovery. That you have to know about it because everyone is looking for chat GPT, AI tools, right? Now you can replace your talk, you can, if you have some feeling, but if you are not able to explain it, so please use chat GPT, right? So drawback of applying AI, it one first expensive. The launch of AI costs hug money consumption, complex designation of machine, maintenance and repairing are highly costly. For this, designing the AI machine, a long period of time is required by the R&D division. And AI machine needs updating the software programming regularly. If you are changing any parameters, we have to completely change the software applications and all, right? So this is the one drawback. Second one is no replicating humans. This is for all of you. Don't be confused, don't be agitated. So robots with the AI technology are associated with the power of thinking like human and being emotionless, as they add some advantage to perform the given task more accurately without any judgment. Right? There is no judgment. Whatever we have put in, accordingly that particular software will give result. No improvement with experience. So human resources can be improved. And there is a value of old and experienced person. They can solve your problem, but software cannot, right? And the last one is no original creativity. Machines cannot do creativity, right? Whatever we have put it in the memory, that machine only work on that particular direction. Next, please. Actually, I was talking about the adverse reaction in pharmacovigilance, but due to limited time, right, sir, I have to close this. Huh? So, in future, definitely we'll talk about the pharmacovigilance. Next, kar dijiye, sir. They will see only. Next. So please be aware about the pharmacovigilance. This is a new era. TCS, Infosys companies are hiring many persons. So please do regularly be updated with the guidelines, whatever we are using for pharmacovigilance and all. Next. Next. How pharmacovigilance come, you know about it. Next. 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 These are some terminology which are associated with the pharmacovigilance program. Classification of adverse event. Next. 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 There are many processes on which basis we can go for the ADR monitoring and all. Next. The vision of everyone being a healthcare professionals must be the world in which people are free from all kind of disease. So thank you everyone. Thank you so much. So once again, I thank you to the dynamic team, Dr. DC Sahu sir, and entire management for inviting me. And really, it was a wonderful conference. And thank you for wonderful hospitality, sir. And it was really nice interaction with all the students. The students were also dynamic and patient. I think you have taken a lot of things on your receptor now, right? So thank you so much. Thanks to all. Thank you.
Thank you so much, Dr. Nitesh Kumar Jain, sir, for your valuable yet mesmerizing word. Now I request respected DC Sahu sir to present memento to respected Nitesh Kumar Jain, sir. Thank you, sir. Now, I would like to request my colleague Vaibhav to join along with me on dance.
Thank you, Sakshi, for having me. I request all the respected dignitaries to please come on the dais. Let's have a big round of applause for our honorable Deputy Minister, Sir Sriram Joshi. I request Honorable Dr. Ajay Bise sir to please come on the dais and share your views regarding the conference. We welcome you, sir.
Respected dignitaries of the dais, faculty members from different institutions, and all your students. This is a wonderful conference which was organized by Raisoni Group. As a regular visitor here, as usually I come for academic audit or as an examiner. So every time I come here, I see a lot of changes and good leadership is there. This theme of the conference was appropriately selected because the current field of pharmaceutical sciences is going in such a way that its applications are not only limited in pharmaceutical or medical sciences, but it is being horizons are being expanded in engineering, pharmaceutical engineering, in aerospace as well. You might have heard there are certain experiments going on with ISRO, and ISRO has a lot of projects, particularly for pharmaceutical field. the effect of drug in gravity, in zero gravity. So they are testing it. Number of their projects are open, and in such type of domain, new domains, they need the new expertise, which is not only limited into the pharmaceutical field, but it needs the expertise from the different sectors, and that's why interdisciplinary, interdisciplinary, and multidisciplinary research approach is a need of today's hour. And very rightly, organizers have selected this particular thing. And I'm very much sure that all those experts from different domains, they have covered all the topics very nicely. The event was very profoundly, very meticulously organized. All micro details, particularly, this was a challenge for organizers that transport of students or the delegates from Nagpur or from different parts to be arranged at this location, and from here again they have to depart it, and they have done it very nicely. That's why there is a really good response. All faculty members, because the success ultimately goes to the organizer, and faculty members, they are struggling or they are working very hard since last 1 or 2 months. And that coordination, that proper coordination among all faculty members, among the different hierarchy of the management is seen here. It has been witnessed here, and this is a wonderful success. And really, all our participants have got a lot of knowledge, and this knowledge will be directing the research domain of the faculty members, of the research domains of the PhD students, because many of the PhD students are here. I've seen them discussing on the different aspects of this. So definitely, this is a win-win situation, a nicely organized platform, nicely organized event. So congratulations to all, and congratulations to the organizing team. Thank you. Thank you.
I request our beloved senior Bhavna ma'am to please come on the dais and share her views regarding the conference. We welcome you, ma'am.
Firstly, I would like to thank Raisoni University Saikheda for giving me opportunity to share my thoughts about this conference. Actually, I was the alumni of this college, so I was not expecting ki. Thank you, GHRU, for organizing such a wonderful conference. It was a great learning here, not only for the poster presentation or oral presentation. Overall conference was very wonderful and very knowledgeable, especially when I was presenting a poster. There was a Nitish Jain sir. He guided a lot, and that was very useful for me. So thank you so much, and it was a great experience here.
Thank you, Bhavna ma'am. Now I request respected DC Sahu sir to please welcome our Honorable Vice Chancellor ma'am by giving sapling. So please, sir.
I'm gonna rock you like a
Thank you, sir. Now, now I would like to call Honorable VC Ma'am on dais to share her words of wisdom with us. So please, ma'am.
Good afternoon, all. Well, I should apologize that I was never part of this two-day conference. Probably I am the last one to join this conference. Due to some reason, I was out of station. But I understand the day all started off with an idea of having an international conference in School of Pharmacy. The people who are from outside, you might have experienced the kind of organizing committee has put down for these 2 days. So, in a nutshell, it was all the teamwork that, along with students and faculties, the show was put up. I hope it was a great learning experience for each one of us. Both subjective as well as those management skills of students pursuing pharmacy. They might have learned a lot. And the feedback that I got today, like I was not in town yesterday, but the moment I came to the office today, I got some positive feedbacks from the participants that they enjoyed a lot and it was truly a learning experience. So I should be thankful to the student committee as well as the staff who have put in a lot of efforts. And there is a lot of women power involved in the staff. So I would say we should give a grand applause to the women power. Because I see the conveners and co-conveners, everyone being a female with one being, being a male staff amongst them. So when we come to an end of a program like this, so what do we take from this? Is many things. Okay, so I'm not a pharmacy graduate to talk about the learnings that you might have got through your presentation or by listening to various papers that were presented. But there are a lot other learnings that one would learn, mainly the networking. People get to know about each other, the student fraternity, the staff fraternity, even the guests. The kind of networking opportunity that comes through with such programs is of immense help. So our journey shouldn't stop at this validatory function or once the event is over, but it should continue. So the people who have visited us or the student of our college should be in touch with the participants who have come across from various colleges, institutions or places. The same goes with the faculty that there were many guests for last 2 days, you had people coming in and going out. So somewhere or another we need to collaborate and move forward. So I think as a whole for the School of Pharmacy, this has been a great turning point, a mileage, a milestone that they have achieved with such a great amount of registration that has happened. And also, they could boast around saying that it is a zero budget program. They didn't get even a single penny from the management to host these 2 days. And in fact, they were a profit center that they could earn and give to the management. And this is what a great learning is all about. This is all about hope, trust amongst the team members that has taken them to this point. I wish the team members, the students, and the participants great amount of luck and best wishes. Thank you.
Thank you so much, ma'am.
It's time for the most awaited prize distribution ceremony. It is with great pleasure that I welcome you all to this prestigious prize distribution ceremony.
We gather to recognize and celebrate the remarkable accomplishments of our winners, who have shown exceptional skills, determination, passion in their pursuits.
This afternoon is a testament to their unwavering dedication and serves as an inspiration for us all. So let us come together and applaud these deserving individuals as we honor their achievements.
Thank you for joining us on this momentous occasion. We have all total of 160 posters and oral presentations.
Firstly, let's head on to the winners of the poster presentation. The second runner-up for poster presentation is Kritika Savarkar from Dadasaheb Balpande College of Pharmacy, Nagpur. I request Honorable PC to please participate for it.
Now she got a 6-year-old trying to keep it
A big round of
I think
applause.
when he looks in her eyes, he don't know he is safe when she said she tells
The first runner-up of poster presentation is Miss Preeti Dongre, Assistant Professor, Taiwan College of Pharmacy, Nagpur.
Congratulations, ma'am. Please give a big round of applause to her. And the winner of poster presentation is Vaishnavi Chaudhary, Medicap College of Pharmacy, Indore. Congratulations, ma'am.
And the consolation prize goes to Harsh Chandak, School of Pharmacy, CHHINDWARA, Saikheda. Now, let's move to the winners of oral presentation.
The second runner-up for oral presentation is Jyoti Trivedi from MIT University, Lucknow. If anyone present on behalf of her to collect the reward, please come to the dais.
The first runner-up is Sakshi Sen from Raisoni University, Saikheda.
The first prize goes to Rita Sayyar, Priyadarshini College of Pharmacy. Congratulations, ma'am. Give her a big round of applause, please. It's time to felicitate all the active attendees present with us from various colleges. Starting off with Central India College of Pharmacy, we have Kavita ma'am, Pragati ma'am, and Vaishati ma'am Ali. I request all the ma'ams to please come on the dais. Please give her a big round of applause. In this conference, we have a total number of 65 participants from Central India College of Pharmacy, Lonara, Nagpur.
Thank you, ma'am. From Dadasaheb Balpande College of Pharmacy, we have Mr. Ajay Vise sir Gayatri Divaskar ma'am, Krutika Savarkar ma'am, and Apurva Tiwari ma'am. So please come on dais. Please give a big round of applause to them.
From the Dharwad College of Pharmacy, we have Ms. Preeti Dongre ma'am. I request ma'am to please come on dais. Please give her a big round of applause.
Thank you, ma'am. Everyone, please be seated. Now, I request respected Dr. T.C. Sahu sir to please proceed the event. So please, sir.
A very good afternoon to all. Dear participants, estimated judge, guests, and honorable BC ma'am, respected registrar sir, Dr. Sriram Joshi sir, a very good afternoon to all in one. Today, I am stand at the cumulative of an extraordinary journey of an international conference has brought together some of the biggest minds from across the globe. As we gather here to close the momentous event, it is my dignity or honor. I stand before you today with a heart full of gratitude and profound appreciation for unwavering response and love that have been endured by the participants throughout the nation. It would be absolutely impossible without dedication and exceptional work of our faculty members, dear students, and all my colleagues. It is with great humility that I extend my thanks to each and every one of you for your tireless effort and commitment to our shared mission of academic excellence. As the Dean School of Pharmacy, Raisoni, I am absolutely privileged of witnessing of transforming import that each of you has to our institution, our institute, and broader academic committee. Now, as a small token of appreciation, I would like to invite BC ma'am and Registrar ma'am. Please give a certificate of appreciation to the faculty School of Pharmacy GH Raisoni. I would like to call registration committee.
I call upon the stage the registration committee members, Mr. Manish Bharati, Mr. Satish Bodle, Bhagyashree Meshram, Hemlata More, Pooja Vaure, and Prerna Patil. Please give a big round of applause for their effortless dedication for the international conference.
They can't do it all. Your name is to the shining stars. And you say you're such a party on the shores. I'm just a singer, it's like yours. I got you on no matter where you are. And then I start up making some this I know that I sleep away. Now it's just a kid I'm really sad. He said one day just want me right, so live life you will remember. My father told me when I was just a child, things on the nights
Second important committee is inaugural and decoration committee and the execution of stage events. Mona Bajpayee, Mona Salvatkar. Sorry. Second committee is for the inauguration and stage committee, the execution of stage events. Ms. Mona Gajpiye, Mona Salvatkar, Ganesh Dekar, Abhilasha Yadav, Neelam Nehare. Please occupy the stage. Please give a big round of applause here. Thank you so much. Now, I would like to request our today's guest speaker, Swaroop sir and Jaiprakash sir, to present the certificates of appreciation to our faculty members. The next scientific session was being governed by Mr. Amol Tere sir, Vijendra Fulsare, Sanjay Uchibagle, Hina Karemore, Shubhangi Chaudhary, and Purnima Patil. Please rise, occupy the stage and receive your appreciation. Thank you so much, everyone. The next committee is Food and Hospitality Committee. It consists of Mr. Santosh Rahate, Ms. Deepika Devdani, Tabassu Bano, Akanksha Bhandarkar. I request Dr. Shriram Joshi sir and Jignesh Joshi sir to please give away the certificates of appreciation to our faculty members. The last but not the least, the Transportation Committee, which was headed by Prashant Gayakwad, Raman Chandak, Praful Hire, Ashwini Pantat.
Please.
Now, I would like to request respected PC ma'am and registrar sir to kindly give over the token of appreciation to our convener, Dr. Dhanashree Dome ma'am, and our co-conveners, Ms. Manisha Gavatkar and Anmol ma'am.
I would like to request once again VC ma'am and Registrar sir to present the token of appreciation to our most important pillar of support, Dr. Jignesh Doshi sir, without whose guidance it won't be possible for us. And last but not the least, our respected deans, I request VC ma'am and Registrar sir to please present a small token of appreciation, but it is not equivalent to the efforts which he has taken for this successful mention of international conference. Thank you so much. Thank you so much, VC ma'am, Registrar sir.
Thank you, Anmol ma'am. In closing, I want to express my deepest gratitude and extend my heartfelt thanks to respected VC ma'am, respected registrars for their support and guidance throughout the event. Thank you, and let us continue our shared journey toward excellence and innovation in education. Thank you. Thanks a lot.
Thank you so much, sir.
I request our beloved Dr. Dhanushi Tumne ma'am to propose a vote of thanks.
So please, ma'am.
Let's give a big round of applause to her.
मुझे लगता है आप सभी को अभी बहुत जल्दी है, but this is very essential for us to present the gratitude towards all the team. We worked very hard to become this event great successful. So, good afternoon, everyone. Respected dignitaries on the dais, of the dais, invitees, participants, my dear students, I feel honored to get this opportunity to conclude the third international conference on approach to pharmaceutical insight through interdisciplinary research, that is APIIR 2023. Certainly, these 2 days are very useful, and we all received great opportunities to make aware ourselves the challenge and development made in the field of interdisciplinary research as well as multidisciplinary research. But organizing international conference is a really challenging task. This cannot be possible without a great preparation, wise working, and support from every corner and blessings of God Almighty. So it's time to convey our gratitude to all who have extended their support to make this event very successful. At the outset, on behalf of organizing committee, School of Pharmacy, I, Dr. Dhanushi Tumne, convener of
APIIR 2023 would like to gratitude to our beloved founder and chief patron, Honorable Shri Sunil Raisoni, Chairman of Raisoni Group of Institution, and patron Mr. Shreyas Raisoni Sir, Executive Director, Raisoni Group of Institution. Dear students, they are known for their exceptional leadership, guidance, and motivation. So thank you very much, sir, for your blessings and always support. On behalf of organizing committee, I extend my sincere thanks to our inaugural chief guest, Honorable National President of Association of Pharmaceutical Teachers of India, APIIR, respected Professor Dr. Milind Lubekar sir, for sharing his precious time with us and motivate the teachers and the participants about pharmacy career. His dynamic leadership in APIIR Council is truly showing the mission meets. Your each word is important for us. I'm very much thankful to our guest of honor, Professor Dr. Dr. Prakash Itankar sir, for sharing his experience while working on the research project and sharing the importance of today's about the interdisciplinary research as well as multidisciplinary research. Now, to acknowledge the gratitude to our jewel speakers, respected Dr. Shivam Srivastava sir, Country Manager, Zydus Life Care, Tanzania. Though he is not present here, but I think he is listening me. So thank you very much, sir, for your valuable guidance regarding the career of students in abroad on topic exploring pharmaceutical industry, career paths, a comprehensive analysis of opportunities in India and abroad. And dear participant, I am very much sure you will enjoy this session very nicely. Next, I extend my gratitude to our respected Dr. Sachin Dubey sir, Deputy Director, Head of Formulation, Ignore Science, Switzerland. Sir, thank you very much for sharing your deep thoughts on quality by design approach to the development of biologics. Respected Dr. Jai Prakash Sangashetty sir, Associate Professor at Bhavi Chauhan College of Pharmacy, Aurangabad. Thank you very much, sir, for sharing your knowledge on ICHQ14, a roadmap for quality by design. Next, I extend my sincere thanks to respected Dr. Swarup Lahuti sir, Professor and Head Department of Pharmaceutics, Bhavi Chauhan College of Pharmacy, Aurangabad. Sir, thank you very much. Your talk was on drug crystallization, a novel approach to alter the properties of pharmaceutical ingredient. Thank you very much, sir. We enjoyed your sessions. You shared grateful things on Q4, this quality by design and ICH Q14. Then, respected Hirendra Kumar Gautam sir, Professor and Director, Shirag College of Pharmacy, Muzaffarnagar, UP. Thank you very much, sir, for sharing your thoughts on drug discovery and interdisciplinary approach to uncovering pharmaceutical insight. Thanks a lot, sir. Then, respected Professor Dr. Nitesh Kumar, sir, and he is a professor and head of the Department of Pharmacology, Modern Institute of Pharmaceutical Sciences, इंदौर. और सर आपकी आवाज बहुत बढ़िया है. नेक्स्ट टाइम हम आपको फिर से बुलाएंगे, तब होप सो कि आप हमें और बढ़िया अच्छा सा सॉन्ग सुनाओगे. थैंक यू वेरी मच सर. Thank you to all 12 speakers for their talks. Thank you. You have accepted our invitation and thank you very much for sharing your precious time with us. And because of you, this is possible to make this event successful. Thanks a lot, sir. Your presence has given to the depth content deliberation. Thank you for coming here. Thank you for adjusting your busy schedule and traveling so long to reach to us and gathering your knowledge and experience. I will believe this conference has opened as a new dimension in our association will go a long way and request to all audience to round a big जोर से तालियां हो जाए please for our guest request. I would like to gratitude, deep sense of obligation and humble thanks to our dynamic Vice Chancellor, Professor Dr. Meena Rajesh Pang, for setting us so well. Ma'am, you and your thoughts have enlightened our minds and have shown us a new path. Because of you and your motivation, School of Pharmacy was confident while working on this conference. So thank you very much, ma'am. You go through always to listeners and you are always ready, immediately solve our problems, always when we are approached to you. So thank you very much. Thank you very much, ma'am. So please a big round of applause for our dynamic Vice Chancellor, Meena Rajesh ma'am. मुझे कुछ बच्चों ने बोला कि मैम यू आर एनर्जेटिक, आई एम नॉट सो एनर्जेटिक. आप लोगों ने देखा ही होगा कि मैम के वर्ड्स और मैम, so we are motivated because of our Vice Chancellor ma'am. So next, I am grateful to our registrar, respected Shriram Joshi sir, for his active support and guidance. You have given your valuable time in fixing many resources related to this conference. So once again, thank you very much, sir. I extend my sincere thanks to Dean, School of Pharmacy, Dr. D.C. Sahu sir, for his active guidance and support. Sir, your mentorship and your encouragement really appreciated. I'm very much thankful to our CIRD Director, Dr. Jignesh Joshi sir, for his active support. My sincere thanks goes to our Chief Finance Officer, Mr. Dheeraj Moryani sir, for his financial support. So again, मैं आप लोगों को फिर से रिक्वेस्ट करूंगी कि थोड़ा सा I take this opportunity to thank all reviewers, Scientific Technological Program Committee for providing their valuable comments in time help towards the improvement of quality of papers presented in the conference. Also thankful to all authors who have submitted their valuable research to this conference. Equally, on personal behalf, I am thankful to all my colleagues of School of Pharmacy, sitting here, coordinators, organizers, for their dedication to make this event to such a successful. I extend my gratitude to registration committee, transportation committee, stage decoration committee, food and discipline committee, scientific committee, and the coordinators and head of this committee are Professor Satish Bodle sir, Manish Baheti sir, Professor Prashant Gaikwad sir, Prashant sir, thank you very much, and Dr. Raman Chandler sir, Professor Mona Dashmiya ma'am, Professor Mona Sarvatka ma'am, Professor Deepika Devanani ma'am, Professor Santosh Rahate sir, Professor Amol There sir, thank you very much. Professor Sanjay Uchibagle sir, and Professor Vijendra Fulsare sir, Professor Ganesh Devkar sir, thank you to all my colleagues. Thank you very much. I'm very much thankful to Bhageshri ma'am, Bhageshri Meshram ma'am, Abhilasha Yadav ma'am, Hira Karemore ma'am, Hemlata More ma'am, and thankful to Professor Tabassum Bano ma'am, Professor Praful Hire sir, Shubhangi Chaudhary ma'am, Neelam Nehare ma'am, Purnima Patil ma'am, Akansha Mandakar, and Pooja Bavre ma'am, Ashwini Parbat ma'am, and Prerna Patil ma'am. I'm very much thankful to all. And the next non-teaching staff पीछे से काम करता है, इतना नहीं है. मैंने सबको रिक्वेस्ट की थी, उद्धव सर को और सभी को कि प्लीज आप गैदर होना सेमिनार हॉल में. So I'm very much thankful to non-teaching staff, Uddhav Bhandarkar sir, then Pushra Shingole, Vijay Kambe, then हमारी वर्षा मैम, वर्षा मदनकर मैम, then thank you to दाते सर और हमारा अभिनव, thank you very much. My sincere thanks goes to, अब इनके लिए तो मेरे पास शब्द नहीं है, to our administrative officer, respected Sanjay Das sir, Anil and his supporter, and to Ablankar sir for the floral decoration and their supporters. Thank you very much. Thank you very much to all. My gratitude to the बहुत बड़ी लिस्ट है, लेकिन आपको सुनना होगा. बस last few minutes. My gratitude towards our store head in charge, आशीष मोहतकर, क्योंकि dear participant, काम करते समय इन लोगों का बहुत सपोर्ट मिला है, तो मुझे तो इन सभी के लिए gratitude feel करना, this is my job. So thank you very much, आशीष मोहतकर. Then आशीष मोहतकर सर ने हमें जब भी कोई रिक्वायरमेंट लगी बस बोलते थे कि मैम इवेंट रेडी होना चाहिए, और हर वक्त उन्होंने हमें मदद की है. So thank you very much. Thank you very much for electrical and technical support to Ashish Gadmande sir and Sarang sir, Navneet is here, and all supporters. I'm very much thankful to Designing Global for providing us designs and printing whenever by adjusting the time, और जब हमें जरूरत थी तब हमें सभी चीजें मिल गई. So thank you to all. So this is all a teamwork. So thank you very much. Thanks a lot. Our last but most important, my special thanks goes to our student volunteers. So student volunteers, these are our shoulders. इनके बिना हम काम नहीं कर सकते थे. So student volunteers, please gather in front of this stage. Please, all student volunteers, please. This work selflessly for this. Yes, selflessly for the success of the third international conference APIIR 2023. Please give them a big round of applause. Please, मैं चाहती हूँ कि हमारे स्टूडेंट को आप फिर से मोटिवेट करें. Dear participants, I express my gratitude to all participants and delegates to make this international conference successful. Without your participation, this success is not possible. So thank you very much, all participants, delegates present here. बहुत लंबी लिस्ट है, लेकिन मेरे शॉर्ट में बोलना चाहूंगी. हमें जो पार्टिसिपेंट मिले हैं सेंट्रल इंडिया कॉलेज ऑफ फार्मेसी से 65 पार्टिसिपेंट हैं. दादा साहब फार्मा कॉलेज ऑफ फार्मेसी से 96 पार्टिसिपेंट हैं. अरुण मोडगरे कॉलेज ऑफ फार्मेसी 15 participants. Guru Nanak College of Pharmacy, Nagpur, 22 participants. Institute of Pharmaceutical Education, Research, Gurgaon, Wardha, Mega College, 25 participants. JK College of Pharmacy, Bilaspur, Chhattisgarh, 14 participants. Kamla Nehru College of Pharmacy, Nagpur, 32 participants. KIT Group of Institution, गाजियाबाद 10 पार्टिसिपेंट. बहुत लंबी लिस्ट है. मैं बहुत शॉर्टली आपको बताना चाहती हूँ कि यहाँ पे लखनऊ से, संगमरमर से, अब्दुल कलाम यूनिवर्सिटी, अजमेर राजस्थान, कोटा राजस्थान, और पुणे से, धुले से, मलकापुर से, नैनीताल यूपी से, सांगली से, from all states of India, we got the participation from participants, and thank you very much to all participants. Near about total college of participants were from different colleges, that is 63, total 650 participants participated in this international conference. In this participant, all faculties, research scholars, and the students are present. So thank you very much to all and present here, and to make this event very successful. At the end, I bring my sincere apologies to those who directly and indirectly help us, and if I forget unwillingly to take their names, so please accept. So hope we all will meet in next event again. As well. Thank you very much. So this is all the people. Thanks a lot. Thank you very much to you all.
Thank you so much, Sanisha ma'am. Now, on this concluding note, I would like to request everyone to raise up for the national anthem and conclude this international conference.
अधिनायक जय हे भारत
तव शुभ आशीष मागे गाहे तव जय गाथा भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Registrar sir to have one group photo. Last 5 minutes. And I request the participants to collect their certificates from the registration counters. And there is provision of item in the corridor. Please take. I once again repeat the participants to collect their certificates from the registration counter and please take the high tea organized for you people. Thank you.
Everything's impossible and you don't get right. You climb around and make the most of you. Don't try, you'll never fly. But you gotta be ready.